Ya cuando quieras, Miguel Ángel.
Okay. So good morning, everybody. Welcome to this online seminar on mechanobiology. So these are the Madrid Mechanobiology seminar series that we started actually already 6 years ago in September 2014. And this was an initiative from the CENIC, the National Center of Cardiovascular Research, where 4 scientists, Jorge Alegre Cebollada, Nadia Mercader, who is now in Switzerland, in Bern, in the University of Bern, and María Montoya and myself, coming from different angles of physics and biology, most of us from biology and Jorge more from a physics point of view, we decided to give a boost to the Madrid mechanobiology community by organizing these seminars. And they have been running for 5 years in CENIC monthly. So every month we will have a seminar of half an hour on biology and half an hour on physics, and then a very lively and entertaining and very nice discussion. And then like a year ago, we decided to give a small turn to increase the interest in the seminars by having the seminars outside CENIC. So with the idea that is now working for like a year is that we are having these seminars every quarter, every 3 months approximately. And we are joining the seminars of these 3 months in only one day, like one morning, for example. And we are having a keynote speaker. Today we're very fortunate to have Thomas Scheibel from the University of Bayreuth in Germany that will be properly introduced by José Pérez Rigueiro in a few minutes. And then we are having a series of short talks. And then after that, we will have a lively discussion, a round table, if you wish. So this seminar was actually planned for spring, for May or June last year. I mean, this year, sorry, it feels like a long time, but it's been this year. Because of the pandemic, obviously we have to cancel because this was a physical seminar. They have been physical seminars so far. But Gustavo and Pepe were strong enough to keep our keynote speaker, Thomas Scheibel. So we are having the same seminar, but online. And I have to say that I think that one of the positive, you have to think positive even in this pandemic situation. And one of the positive things of this is that now we are having these seminars online and open to everybody. So I think that this will be very interesting if we can keep it. And at the end of the seminar, we'll have some words about that. I want to thank everybody in CENIC that is organizing this together with me, Jorge, Maria, previously Nadia. Unfortunately, for different reasons, I don't think that they can be here today for different reasons. But they will be obviously in following occasions. So I also want to mention that this started with this change in having the seminars outside CENIC. It started with Fernando Moreno that organized that last year in spring in CMB, and then Arrate Muñoz, who did the same in the University Carlos III de Madrid. This was like February this year. And now, you know, I give I want to obviously thank Gustavo, and Maite and Pepe and everybody in the Center for Biomedical Technology and the Universidad Politécnica de Madrid in the campus of Montegancedo. That is the physical place where we plan to have this seminar. And they have been responsible for all the hosting this event today. So without further ado, I really welcome everybody here. We are having many people, and we are hoping to have more. And I give the word to José Pérez Rigueiro to chair the plenary talk by Thomas Scheibel. Thank you very much, and welcome, Thomas. Thank you for coming.
Well, thank you, Miguel Ángel. First of all, I would like to thank Professor Scheibel for his willingness to participate in this seminar under these very complicated circumstances. And I would like to start with some biographic details of Professor Scheibel. Professor Scheibel is full professor for biomaterials, as well as vice president for internationalization, gender equality and diversity at the University of Bayreuth. Since 2014, he is a member of the German National Academy of Science and Engineering. He is also chairman of the Bayreuth Melbourne Colloids and Polymer Network, co-chairman of the Trans-Regio SFB Biofabrication, member of the review board Biomaterials of the German Research Foundation, advisory board member of German Association for Resource Efficiency and Additive Manufacturing, co-founder and shareholder of Silk GmbH, group leader of New Materials Bayreuth GmbH, and director of Bayhost in the Bavarian Academic Center for Central, Eastern, and Southeastern Europe. And I would like to add that Professor Scheibel is one of the world leading researchers in the field of silk and recombinant silk and application of silk. And he is also a person that has the ability to convey his knowledge in a very inspiring and motivating way. And so I'm sure that we are going to enjoy a most interesting and formative talk. So Professor Scheibel, when you wish.
Thank you very much, Pepe. So it's really a great pleasure. Be again in Madrid. Of course, I would love to be there in person. I miss that place. I've been there several times. I always enjoy the hospitality. I enjoy the science. I like the city. I like the football teams. And tonight there will be a match between Bayern Munich and Atletico Madrid. So I'm looking forward to that too. But of course, now I'm pretty much eager to learn about your science. And before I learn about your science, I will introduce you a bit into what we are doing. And I will share my screen now. Okay. So can you see the screen? Everything's good?
Everything is good. Yes.
Yes, everything.
Okay, thank you. Yeah. So together with Pepe, we decided to give you a brief insight into what we're doing in two areas of biomedical research. One thing is providing materials for heart muscle tissue regeneration, and the other focus today will be on peripheral nerve regeneration. And I will show you what we actually can do with our types of materials. But before I come into these details, I would like to actually introduce silk a bit in more detail and about our recombinant silk technology. So if you have a look at silk materials, and we are specialized in spider silk, but you can also expand that to silk from silkworms or other insects. Many silks are suitable for biomedical applications, and the reason for that is that silk is a protein-based material, so it's biodegradable, as you can see up here. You can actually use protein-based materials to load and release biological factors, which is a benefit for wound regeneration or any kind of tissue regeneration. We can implement binding sequences like the very well-known integrin binding sequence RGD in order to actually get a cell interaction with the silk surfaces. Silk are very well known for their proper mechanical properties. They actually do not show immunoreactivity inside the body, and this is very surprising. But a huge benefit of using silk materials over other materials, since the human body obviously does not recognize silk as some foreign material and it does not react towards it. And in that context is also the lack of toxicity, which means we can actually really safely use silk materials inside the human body. I'll come back to that in more detail a bit later. So if you look at the natural sources of silk, as I mentioned, we are focusing on spiders, but there's also a lot of other arthropods, mainly insects, that are also able to form silks, like the very well-known silkworm, Bombyx mori, but also bees or wasps, lacewings, they produce silk. And they produce silk with different properties. And this is very important to know, that silk is not silk, but there's a huge variety. All of them are made of proteins. And if we have a close look, we see the different silk structures that can be adopted. The common theme between spider silk and silkworm silk is the presence of beta sheet crystals. And they have a huge impact on the mechanical properties, as you can imagine. In the bee and wasp silks, we see a lot of helical silks, so completely different protein structures. And of course, then these silks have different properties. And the same is true for the cross beta silk that is made by lacewings. So you see here 3 different types of structures, and obviously they're used for different purposes. And you see the purposes underneath. The world experts in silk production very likely is a spider, because spiders can use multiple silks, up to 7 different types, for producing webs, making cocoons, nests, and so on. So they are the specialists, so they can actually decide which type of silk they use for whatever application. In silkworms, the silk is mainly used for making the cocoon, so protecting the offspring during the transformation from the worm into the butterfly. Wasps and bees, they use their silk for housing, so they actually make composite materials together with wax to make their nice housings where they live in. And lacewings use silk, for instance, for breeding, so they produce egg stalks. And the egg actually is protected at the tip of these egg stalks from predators. So you see different types of applications: prey catching, offspring protection, housing, breeding. And this comes along with different properties that you need for these applications. Cross beta sheets, like the lacewing egg stalks, they are bending stiff. Yeah, this comes from this explicit structure. These helical structure, this allows really the incorporation into composite materials with waxes, because here you can easily provide hydrophobic surfaces that are necessary to interact with the wax. And we will focus now here on the beta sheet structures as we find in spiders and silkworms, and the main focus will be on spider silk. This is what we're working with, and just to give you a very brief introduction into spider silk, you see here a badminton racket, a speed badminton racket, and we have here spider webs just turned in, and you can play with this racket. And this is very surprising, because the individual fibers, they're really thin, so they're between 3 to 5 microns, and this gives you at a first glance already an idea that it's mechanically strong. And this is obviously a natural property, because the web has to be strong to actually hold up the impact of the inflying breath. Then, if you have a close look at the surface, you don't find microbes. So it's obviously somehow bacterial, repellent, and I come to that later in more detail, because we just had recently a publication on that topic. It's well known since centuries that spider webs can support wound healing. They do not cause inflammation, and they do not cause allergic responses, at least not to our knowledge. So combination of really excellent properties, especially if you think about biomedical applications. Now, I told you already that spiders are experts in making silk because they have huge variety of silks, but there's also a huge variety of spiders. So far, more than 48,000 spider species exist, and we just recently identified a very new species. One PhD student of mine found this species in Colombia, and she had the privilege to name it, and she named it after her German language teacher in school. So we are now very happy that we also identified a new spider species, but this is not our main topic. Our main topic is the silk, and as you can see, different spiders, they produce different type of webs. Yeah, they not all look like this web. Sometimes they are actually located in such a boat, wooden frame. Sometimes they're free hanging. Sometimes they're a bit like messy, 3D, 2D, whatever. So you see different types of possibilities how to deal with the material, and obviously all the materials will differ slightly. And especially the Madrid group is very well known for mechanical analysis of the different silks, and they found out that indeed there are differences in the silks by different spiders, and this of course makes to me a lot of sense because they're used for different kind of applications in different kind of economic niches. So are spiders dangerous? I'm always asked that. And here is a sign that I found in Australia in the Brisbane Museum in Queensland Museum in Brisbane. This is the cause of death in Australia in 2013. And you know that in Australia, there's the most toxic species, and there's also the most toxic spiders there. But are they dangerous for people? Well, if you look at the numbers in 2013, roughly 44,000 people died of cancer, 30,000 on heart disease, 2,000 just fallen off a tree or a ladder whatsoever, 700 in car accidents, and so on. 4 were bitten by snakes, 2 by sharks, and 0 by spiders. So spiders are not dangerous. Spiders run away. So whenever you approach a spider, it will not attack you. So don't believe what you see in B movies, yeah, so that the big tarantulas go after you. No, they're not. So we are not their prey, and they try to avoid us. I'll promise you that. So if you look at the classical orb weaver, so these are the spiders with the most different types of silk, like the European garden spider, and this is one of our working horses in the lab. You see that indeed they can make up 7 different silks. So they start with the frame, which is the so-called major ampulate silk. And then actually this is accompanied by the capture spiral. This type is called flagelliform silk. This is just a mechanical setup. In order to keep the prey in the web, it needs some glue, and this type of spider uses a liquid glue. There's also other spiders, they have a dry adhesive wool-like structure, but these spiders here, they use a liquid glue, which is called aggregate silk. And this actually manages that the prey stays in the web as soon as it's getting in. Then there's the supporting silk, the minor ampulate silk. There is something to attach it to the surroundings. And then you see additional silks here for egg case shielding, for prey wrapping, and so on. 7 different types of silk, all have different properties, as you can imagine: sticky, aggregate silk, not sticky, the other silks. I will focus on the major ampulate silk because this is the best known silk. We not only work on major ampulate silk, but everything you hear from now on will be about the major ampulate silk. So the material has extraordinary properties. Why don't you see applications for it? Well, that's not true anymore, because I will show you that indeed there are applications there, and there are commercial products available for everybody. I will come to that in a minute. So first of all, I would like to introduce you our technology, because there is some problem with spiders. They are cannibalistic. And we call the technology Spider-Man technology, and I know Spider-Man in person, and you will see that in the movie. Oops, I have to go back. If I can start it, yeah. Yeah, so this was my son. What is the problem with spiders? Well, the problem with spiders is they're not a problem for us humans. They're not killing us, but they killed themselves. So most spider species are cannibalistic. So it's a big problem to actually keep a lot of them at a small place. In order to get a lot of material, you need millions and millions of spiders and a lot of co-workers, and this is a very expensive feature. So therefore, for any commercial product, using the natural material, using natural spider silk is not a good idea. In order to circumvent that, you can think about biotechnology. And biotechnology started in the 1980s to produce proteins in different hosts. We call that recombinant technologies. So you transfer the genes from one organism to another organism, and this other organism produces then the proteins. This is also a bit tricky because all genes are encoded with a codon usage, and this is different in spiders in comparison to typically used hosts in biotechnology, in bacteria. And therefore, we had to actually get around that. And what we did, well, we took the spider silk genes, we put it in a plasmid, and then we changed it. We made engineering, molecular engineering, to adopt the codon usage, to make it a bit better for the bacteria. Then we actually introduced this plasmid into bacterium, and then we can do large-scale fermentation. And this runs now in huge facilities. And actually nowadays, companies can produce up to hundreds of tons of spider silk proteins just by using bacteria. The point is the bacteria only produce the molecules, right? They do not produce fibers. So this is another big question. But as mentioned before, this is not the main topic, but I wanted to show that indeed you can nowadays buy products for everyone. So this armrest for these watches is the very first full 100% protein fiber scaffold. So this is actually made of our spider silk proteins. You can buy that through the online shop of Omega. And there's a lot of cosmetic products because even without having fibers, you can use silk to get smoother skin, better hair, whatsoever. And you see a lot of products that you can buy even here in the drugstore, in the pharmacy, and in Germany, it's available everywhere. And I think some of the products are also available in Spain. We sell them to South America, South Africa, Australia, Asia, and you can buy these products everywhere. So nail polish. So this is a tick removal coating. This is for coloring your hair, skincare, whatsoever. A lot of different shampoos, a lot of different things, which actually has our technology inside. But this is not the topic. Now I would like to focus on medical applications, and this is what we at the university are really interested in. And as I mentioned, the bacteria, they produce proteins. So what we have to do, well, we first have to process these proteins, and this is where now the engineer side comes in. So far we were molecular biologists, right? Now, actually, we get into the details in processing. And the very good news is that you can process spider silk proteins pretty much into whatever you like. So this behaves like classical polymers. So you can make fibers, of course, as in nature, but you can also make capsules and forms and nonwoven meshes, hydrogels, films, particles, and so on. We could show that more than 10 years ago that it's possible to make all these different types of morphologies. But the big question, of course, is A, do biotechnologically produced silk proteins have the same features as the natural spider silk? And B, what happens if we process them? Do we change then maybe some properties? And in other words, are all these morphologies also biocompatible? So what you have to do is to get into more detail, you have to severely test the body response. And I can assure you that no matter what we tested, what type of morphology in whatever normed test, we showed no immunogenicity, so no antibody response, no toxicity, no skin irritation, eye irritation, mutagenesis, and so on and so on. So it's a very safe product, and it was CE certified in 2014. So this actually allows us, since more than 6 years, to actually use it inside the human body. And the first product will come on market probably within the next 2-3 months. So we have all the approvals now at hand. So let's see what happens if we make a silk surface. Best to investigate is a flat film and see how human cells like it. Well, first we started not with human cells, we started with mouse cells, mouse fibroblasts, bulb-free T3 fibroblasts. So these are skin cells from mice. And to our surprise, so this is a tissue culture plate, and this is the silk fiber, the silk film. We saw that the cells don't like to stick to the surface. So if you make a proliferation test, so if you look over days, what happens with the cells, you see that they actually, they agglomerate, they still are in a spherical shape, they're not attaching the surface, they're not spreading out, and they're not proliferating on the silk surface. But they do, of course, on the tissue culture plate. This was a very first indication that in this state, so in the native state, the spider silk is probably pretty much like a cell repelling surface. But that's not bad, because sometimes you want to have that inside the human body, that the body cells cannot recognize the surface. So we thought this could be a clever idea to protect implants. And this was the very first setup many years ago. And you see here, the publication was in 2014. And now the product comes on market because these are silk-coated silicone breast implants. How to coat a silicone breast implant? It's quite easy. You just take this implant, you dip it in a silk solution, an aqueous silk solution, I have to say. And silk molecules are amphiphilic. So what happens is they immediately assemble on the surface and they make about 1 micrometer thick film. So we get then a shielding, a coating of this implant. And to make a long story short, well, first we tested that in vitro, then we tested it in vivo in rats. Now we actually went through the complete clinical trials with humans. And there is a lot of patients already in Europe running around with our implants. And therefore we get now the approval to get it freely on market, and this will start soon to get it on market. Here you see the benefit. So this is just a plain silk silicone implant, and this is the silk coated implant. And you see a clear difference, even here in rats, that silicone surfaces, they're very attractive for human cells and for rat cells in this case. So what happens is the cells go to the surface and they would like to destroy this foreign material, but silicon is pretty strong, so it's not biodegradable. And what the cells then do, they start to encapsulate them. So actually they start to make cartilage surrounding this implant, and they start to make a capsule. For the rats, that's not a big issue. For human beings, that can be an issue, especially if you're a breast cancer patient, because you have a depressed immune response system. And therefore, these capsules can be really causing severe side effects. Because what might happen is an inflammation, and in about 25% of the breast cancer patients that got a silicone breast implant after their surgery, this implant has to be removed because it causes problems, and this is of course not nice for the patients. In contrast, if you look at the silk-coated implant, you see there's no coating. There's actually just tissue growing around. You even see vascularization, and this makes this coating very, very successful. Here you see what happens with the cells, and here we just had some DAPI staining, which stains the cell nucleus, and some collagen 1 staining after 7 days of cultivation. Uncoated silicon, you see that here the fibroblasts, they grow, they have a lot of collagen production. The silk-coated silicon, as before, they're not even spreading out, they're not proliferating, they're not making collagen. And this is the explanation why we don't get capsule formation and we don't get fibrosis. So we tested a lot of factors in first in rats, then in humans, and you see that most of the inflammation factors go down. So this is always in comparison with the non-coated silicone implant. You see that a lot of factors, they're dramatically reduced. There's only one factor, the CD4, that is slightly increased. But if you look at the factors, all the others are negative, which means they're all much lower than in the uncoated control. Inflammation factors, fibrosis factors, TGF-beta1, which is a very important marker for capsular fibrosis, severely reduced. Contracture markers, fibroblast to myofibroblast transdifferentiation. So this is an indication of wound healing, pretty much reduced. So we have a long list, this is a very, very positive, because this led us to the model, which actually was now confirmed in human beings, that if you have a silk coating, and this is here just done for make it much clearer, because obviously we will coat the entire implant for the real stuff. But here we just coated half of the implant to show you the differences, what happens on the surface. On the silk surface, we have no cell proliferation, reduced extracellular matrix synthesis, no fibrosis, reduced contracture. So this looks much nicer than the other part. And this nicer surface, of course, has the benefit that we have less severe side effects. We don't see any fibrosis, for instance, in the patients. Now let's go back to the main topic. In the title, I promised you to talk about heart muscle tissue repair. So big question is, can we repair a broken heart, so to say? Yeah. What happens if the heart muscle is damaged? And this is a very common thing. Just in Germany, we have annually around 7 million heart attacks. Not all of them might lead to death, but what all heart attacks do, they block some of the blood vessels in the heart muscle. So the heart muscle is not innervated anymore, it doesn't get oxygen. And this actually leads to the death of cardiomyocytes, so the heart muscle cells. And cardiomyocytes have one drawback. They cannot migrate. So whenever you have a damaged zone, here shown in yellow, heart muscle cells cannot re-enter the zone and they cannot regenerate the muscle. And therefore, we always have scar formation. And you might now be able to imagine that if you have a scar in the middle of a muscle, you will have a severe impact on the function of this muscle. Yeah. So even if you survive the heart attack, there is some side effect because the muscle is weakened by this scar that is right in the middle. Yeah. So the idea is to support the heart muscle. So far, it's done by patches. So something is put on the top of the muscle to support the contraction. Our idea now is, can we actually really replace this scar tissue? So remove it by surgery and replace it with a real regenerated heart muscle tissue. That's the idea. So for that, we have to overcome some problems. One problem is to find the right material. We need a scaffold material where the heart muscle cells really like to be on. And one such material is fibronectin. And with fibronectin, this works very nicely in 2D, as you can see here. So heart muscle cells, the cardiomyocytes, they like fibronectin surfaces. They spread, they connect, they communicate. One problem with fibronectin is you only can do that in 2D. There is so far no technology available, and fibronectin is probably not the proper material for that, to make 3D scaffolds. But we need 3D if we want to regenerate a muscle, right? So therefore, we actually investigated silk, because as mentioned before, we can do with silks whatever. We can even 3D print that. Yeah. So the main question here was, does cardiomyocytes interact with silk surfaces? And to make a long story short, again, with our regular silk, they don't. Yeah. Remember, no cell would like to attach to this silk. So therefore, we had to actually further engineer our silks. And what we did, we introduced now positively charged amino acids. So on purpose, we actually changed the surface charge of the silk, which is typically negative, into a positively charged surface. The rest was the same. And out of a sudden, so we call this protein E80F4 for engineered Araneus diadematus, so that's the spider, fibrin 4, so this is the natural counterpart, kappa 16. Kappa stands for K for lysine, and we have 16 lysines in there. And if we make this engineered spider silk protein, you see that the cardiomyocytes now out of a sudden, they attach to the surface, they spread, they proliferate, they connect, and in the movie you see they even start to contract after 2 or 3 days. And we even could keep now beating cell cultures, cardiomyocyte cultures, for more than 60 days in culture. So as soon as they start beating, they're very happy, they like it. However, this is 2D, yeah? So, but this was the very first important step, to find the right material. The question is, can we also use other silk engineering? And as mentioned at the very beginning, we can introduce also cell binding motifs like the RGD. So we tested again now, so this is our working horse, the negatively charged protein, E80F4, you know what that means. C, this is glutamic acid, so they have a lot of negatively charged groups. And now we added the sequence RGD. And you see now all of a sudden, well, this surface is also good for cardiomyocyte adhesion. And even if we increase the concentration of the fetal bovine serum, you see much nicer proliferation. And again, make a long story short, cardiomyocytes start to communicate, and they actually start with a beating rate, and the beating rate is even higher than on fibronectin. So the white bars, they are with spider silk, the black bars are with fibronectin, and here you see the beating rate in minutes. And why is that important? Well, it's not just important that they beat, they have to beat in the right rhythm. And so we see here an increase in the beating rate already in 2D, and this is a very good sign, because this actually tells us the spider silk variant is even better than fibronectin. So if we now look for this first step, we have to go now further ahead, because we have to go into 3D. And if we want to have a really 3D working muscle, what do we need? Well, we need vascularization. So we need blood vessels to grow into the silk. And for that, we hooked up with a team from Erlangen, and they are specialists in a so-called AV loop model in rabbits. AV stands for arterial venal loop. So actually they cut open an arteria and a vein, and then they artificially connect it, and they put it here in such a Teflon pan. So you see here, actually the arteria is coming in, and then it's going over into a vein, and it's in a Teflon pan. And inside the Teflon pan, now you can place a lot of materials. And if they are actually good materials, the vascularization from this AV loop into the material will start. And here we tested our spider silk. It's typically in the rabbits for 2 weeks and for 4 weeks. And then it's checked if after 2 weeks or 4 weeks, any new blood vessels are grown. So this is how the explant looks after 4 weeks. And this is what happened. And to our surprise, we see a lot of vessels growing out of the AV loop. And in this histological picture, you see a lot of newly formed blood vessels. All the black dots, these are new blood vessels that have been formed. So they're artificially colored to make them visible. And all the black dots, this is new material. These are new vessels that have been formed inside the silk material. So next step is done. Actually, in vivo, our material will be vascularized. And this is very important, because if we have no blood supply to the cardiomyocytes, they will die. So we need that to take place. Another big issue is infection, and this is a study that has just recently been published in Materials Today. Of course, in all these tissue engineering approaches, we always have the problem with infections. We have a lot of pathogens we have yeast, we have different bacteria, we have multi-resistant Staphylococcus aureus, and so on. In our respiratory system, in our intestine, everywhere. Yeah? So we're flooded with microbes. And of course, they cause problems if we would like to make tissue regeneration. So what we need is to prevent them from growing into any new kind of tissue. And typically this is done with antibiotics. But as mentioned, some Staphylococcus aureus, they are already resistant to most commonly used antibiotics. And also you cannot give antibiotics forever, right? So what you actually will need is a microbe repellent scaffold material. That would be the best. And you can do that by structuring surfaces. Yeah? Nanostructuring of surfaces has already been shown to be microbe repellent in comparison to microstructured surfaces, because they are actually well suited for bacterial adhesion. So nanostructuring is the key, but nanostructuring will also repel, again, all the human cells. So we are again in the problem. If we generate a nanostructured surface, we would like to have now bioselectivity. We would like to have the body cells growing on top, but at the same time having the bacterial repellency. And this was something that we investigated, though the whole study took 8 years, no kidding, 8 years to get the results, what we can do with spider silk. And here you see, again, our working horse, our negatively charged silk, flat films, and structured films, because we wanted to find out whenever we have surface topography, if this might actually accelerate or support bacterial growth. And here you see one of our control materials, which is polycaprolactone, a very often used polymer in biomedical applications. Here we just tested bacteria like Staphylococcus mutans, and down there we actually tested a yeast, Candida albicans. And as you can see, nothing is growing on the silk surface, no matter if it's a flat surface or if it's a structured surface. In contrast, on the polycaprolactone, you see actually huge areas covered with bacteria or with yeast, and this is what we call a biofilm. So we see immediately after a few days biofilm formation. So we already could show that on flat surfaces, spider silk works very well. And this, of course, accompanies what I've shown you before with the fibroblast studies and with the silk coating of the breast implants. Nothing likes to stick to it. So now the table is set. So we have a material that is bacterial repellent. We have some engineering in the silk that allows us to attract human cells, specifically the cardiomyocytes. We know this works in 2D. The next step is to go 3D. And for that, we have to go now to silk hydrogels. And this has been shown almost 10 years ago that we can make hydrogels out of spider silk. And this is just some SEM pictures to have a glance inside, and we see a porous structure, and the pores are fairly big enough to actually harbor human cells. So we thought these hydrogels are very well suited. The next step was, can we actually give these hydrogels also specific shapes? And for that, we tested robotic dispensing. So what you do is you put this hydrogel in any kind of capillary, and then even pneumatic or with a piston or with a screw, you actually press it through a needle. And if you have a good hydrogel, it starts to flow when actually the pressure is set on, and it immediately hardens if it comes out. So this is called the shear thinning behavior. Yeah. And then you actually can make precision 3D scaffolds. If you have a good bio ink, this is what we call a bio ink, that has this behavior, that it's hard without any shear force. You press it, it starts to flow. Then it's good bio ink. And you know this behavior from toothpaste. I mean, toothpaste is not perfect because it also shows some instabilities later on, but in principle, if you have toothpaste in the tube and you open the screw, nothing happens. You have to squeeze it that it flows, right? And that's pretty much the same principle here. Okay, so can we do that with spider silk? This was the first test, and you see here a 3D printing in real time. So you see it immediately hardens as soon as it leaves the needle, and you immediately can start with the next layer. Mm-hmm. And the first idea was, well, now we 3D print the stuff, and here we 3D printed something that looks like an ear, 64 layers, and then we actually inoculated cells on top. The drawback of that system is, and this is for all kind of tissue engineering approaches, that the cells have to go into the scaffold and find the places. So especially with the cardiomyocytes, I've shown you before, they're not migrating. So if we just would put the cardiomyocytes on top of such a scaffold, they will never enter it. They just will sit on top. So therefore, we had to actually overthink that kind of story. And the idea was very simple. Well, if we just place the cells together with the materials in the scaffold, that would be the best option. And this is what is called biofabrication. So the simultaneous printing of the hydrogel with the cells. So we mix the cells with our spider silk ink, and then we actually print that together. And out of a sudden, the cells are in place. They don't have to migrate anymore. And the first thing we looked at was, do the cells survive that? Because if you just print cells without any scaffold, they're dead. Most of the cells die. And here you see a live dead testing after 40 days, and you see a lot of green dots, and probably you see non-red dots. A red dot would be a dead cell. All the green dots, they are living cells. So we have a survival rate here in the printing process of more than 99%. So actually the silk gel is not only the scaffold here where the cells sit in, but it also shields the cells during the printing process. So it shields the cells from the shear stresses, from the elongational stress that appears down here at the needle. And this actually allows us now to 3D print cells with the scaffold, and they're surviving that. Now let's go back to our bacterial story. So I showed you that the surface of the silk is microbe repellent. How about now in the hydrogels? Because if you have here a bacterial manifestation, that could be also very severe. And you can see very nicely that, again, our spider silks, and here you see our working horse, the working horse with RGD, they actually see no bacteria or yeast manifestation. In contrast to gelatin, which is very often used in 3D printing and biofabrication, Bombyx mori fibrin, so silkworm fibrin, that does not have these repellent features. And even if we modify this silk here, if we remove the charges completely, omega-16 is uncharged silk, then you see that we lose some of the ability of repellency. But be aware of, here's a break in the y-axis. So this is numbers of magnitudes higher in silkworm silk and in gelatin. And this is how it looks like. So actually what we did, we actually floated these hydrogels with bacteria in yeast. We incubated that for days. And then we just washed it. And you see that both spider silk hydrogels, they're absolutely clear. There's no single bacterium in there. And the silkworm hydrogel and the gelatin hydrogel, they're actually grown over with the bacteria and with the yeast. So what does that mean? Well, this means now we could do co-culture experiments. So we could actually put fibroblasts and bacteria into the scaffold at the same time, then incubate that, and then actually check what happens to the fibroblasts and what happens to the bacteria. And the experiment is shown here. So the fibroblasts, they survive very nicely. So we have around about 95% survival rate over 2 weeks. So the fibroblasts are fine. And again, we have here an axis break, yeah? And look at these numbers. There's undetectable amounts of bacteria in gray, and in yeast. So even in the hydrogel, this system works. What does that mean? Well, with spider silk, especially with the RGD variant, we now have a bioselective material that allows us to selectively grow cells on top and very selectively repel microbes. And we right now look at a lot of pathogenic relevant strands, multi-resistant strands, and so far we have not found any bacterium or yeast that can grow on the silk surface. Well, the story continues, but these are very exciting findings for us. And the reason is silk has a nanostructured surface. Remember, this is one physical principle that has been shown also by others that works in microbe repellency. So we can repel microbes just through the nanostructure on the silk surface. And if you want to find out more, this is published in 2 papers, Materials Today and Data in Brief, and they both combine the study that we took over 8 years, as I said. Last story, I make it short. Can we also repair peripheral nerves? Now, different topic, yeah, different story, but this is also something that we're very much interested in. So if you have peripheral nerve injury, so I'm not talking about the central nervous system, I talk about peripheral nerves. You cut off a finger, yeah, so you cut off a peripheral nerve. So nerves can actually, in principle, regenerate naturally. But this only works if there's a very small gap, yeah? Because you have then loose ends of nerve fibers and they have to find each other again. The further you get apart, the harder it is that they find each other. So therefore, if you have a bigger gap, there's just one probability that you actually get an autologous nerve transplantation. What does it mean? Well, they take some nerve, typically from the forearm, and took that out and replace actually the damaged side with this autologous nerve. The problem is you have a loss of sensibility at the donor side. That's one thing. And of course, you have not unlimited numbers of nerves that you can take out of one patient. So this gives us some limitations. Therefore, a group at the Medical University in Hanover, they started to use spider silk, and they took spider silk from spiders. Yeah, so major ampleted silk fibers from spiders. These were supposed to be guiding structures for the nerve cells. Then they embedded this in matrigel. Matrigel is a gelatin hydrogel where they implemented Schwann cells. And then they put this whole thing in a cava vein of a donor rat. So they removed all the cells from the vein, and then they put the silk spider silk fibers and this matrigel inside. So that was the setup. That's the vein. These are the spider silk fibers, and here in between you see the matrix of matrigel. And this looks good. So they actually tested that also in vivo in sheep, and they could span 60 centimeter defects in nerves with these, what is called a nerve guiding conduit. Yeah, so this is like a highway for nerve cells that they can connect again the loose stumps one to the other. And you see here the neuronal cells that they grow and they connect and so on. One drawback is you need a vein, and obviously you probably don't want to have a rat vein in your body. Matrigel has some problems because there are batch-to-batch variations in the performance. And as mentioned before, a natural spider silk is not that easy to harvest at large scale. So what we try to do is now using our technology, our biotech silk, to make nerve guiding conduits. And we also wanted to have a faster approach. This approach takes roughly 6 months. So if you want to span centimeters, the cells have to grow in, they have to connect, to proliferate. This takes a long time. We want to have a very rapid technology that allows us to actually get the nerve back to function quite quickly. And for that, we thought about self-rolling systems. So if you combine spider silk with chitosan, you have a 2-layer system that is responsive to liquids, because if you actually now add here cell culture medium, the chitosan will expand differently than the spider silk film, and you will get a self-rolling. So you actually can create out of a flat film a tube. And the benefit of that is that before you do that, you can actually implement other things. You can actually implement like spider silk non-woven mats. You can have also collagen cryogels. You can actually fill this stuff with whatever you like. We add the nerve cells, and then you actually have a nerve guiding conduit, which is ready to go within a few days. That's the setup. It also has recently been published. This is how the setup looks like. So you see, that's the filler. In this case, it's a collagen cryogel. And this is the self-rolling spider silk tube. And you see everything is nicely packed and the fibers are aligned inside. And of course, we have the cells in place. Same principle as with the heart muscle regeneration. We put the nerve cells already along the structure. So we don't have to wait until they crawl in and find their place. We put them where they should work. That's the idea. And then you see that the nerves nicely align along the channels in the cryogel or along the fibers, and they get functional. We also can use collagens for that. And this is a technology that we developed a few years ago, a microfluidic setup for spinning collagen fibers. And I wanted to show that to you because this is also a very unique technique to make endless collagen fibers with unique properties. All you need is collagen solved at a slightly acidic pH. You have 2 neutralizing solutions, and then you have the setup where actually the collagen solution is not getting in contact with the wall of this fluidic channel. And then you can make fibers like that endlessly. You can make knots in them, so they're mechanically quite stable, and they're much more stable than any kind of other collagen fibers. We have a higher Young's modulus, we have a higher strength, we have very good extensibility, and we have a low diameter. So these collagen fibers, they can also be implemented in such a nerve guiding conduit. You can even scale that up, and the scale up is running, so we actually can produce now kilograms of that stuff. We can even braid it, we can make new kind of structures for any kind of application. But back to the neural regeneration, these collagen fibers that we produce with the microfluidics, they're also very well suited for neuronal cell growth. And again, the neurites and the axons, they grow along the fiber axis. And here you see them much better. So we can actually pre-align very nicely the neurons with these collagen fibers. Second setup that we used, I've shown you before the self-rolling films. We can also use silk nonwovens per se and roll that up and then actually bring in the collagen fibers. So this setup is more similar to the setup of the Hannover group, but instead of having a vein, a cover vein, we have now here a silk nonwoven, and we have collagen fibers instead of the silk fibers. And this setup also works quite well. We see that the neurons, they actually really make neuronal networks. They interconnect, and you see here the connection points between the neuronal cells along the fibers. And we can even measure in vitro now the signal propagation through these conduits immediately 2 days after cultivation. 2 days versus 6 months. And I think this is a huge improvement. So this is our technology that we would like to bring in place. And of course, this is only done in vitro so far. So the next step will be to do that in vivo and to see if we really can bridge damaged nerves, peripheral nerves. And with that, my time is up. Come to an end by thanking all the people. So here you see my group. Well, this was before Corona. I do not want to change the picture because right now it would be, you won't see the people because they're so far off. I would like to thank all the sponsors of the studies, our companies that support us a lot, our partners, I mentioned some of them along the way, international partners, Bayreuth partners, national partners, our alumni, and finally, I would like to thank you for your attention, and of course, I'm happy to take questions if there are any.
Well, thank you very much, Professor Scheibel, for your justifying my initial remark about an extremely formative and informative presentation. Absolutely. And with regard to the questions and due to the limitations of this format, I would suggest that, well, there might be two options. Either using the chat, and there is at least one question now in the chat, but if somebody wants to ask you directly and to have the opportunity to talk to you, I would also be willing to open the microphone of this person. So there is, as I say, one question right now, and while there are more coming, it's from Manuel Antonio Oria. And the question is, if the silk by itself has a low cellular attachment property and low cellular proliferation, as said previously, how is it possible to create an hydrogel in which the aim is to harbor cells? Is the silk modified in order to alter this property?
Yeah, so very good question. And I hope, so I also see in the chat that my microphone had some problems. We sometimes have, I apologize. So I hope you can hear me now nice and clearly. Yeah, so this is the RGD variant. So we need to have the RGD modified silk because with the non-modified silk, well, the cells will be inside, but they will not actually spread out. They will not proliferate. Yeah, so it has to be modified with RGD.
Well, let's see if there are more questions.
I have a question. Can you hear me? I'm José Jalife.
Yes, please proceed.
Yes, Professor Scheibel, this is a very interesting presentation. It's a huge amount of work, and I really congratulate you for such an accomplishment. I'm interested in the heart, and my interest is in trying to understand how cardiac cells interact with each other. And tissue regeneration has been an important area of research over the last 10 years in the heart for myocardial infarct. And really bringing new materials into the field is a very, very important area of research, which is of huge interest in the field. However, and what you presented to us sounds very, very interesting, but the devil is in the details, as you know. And one of the things that has happened in the field is that even though some investigators have been able to introduce cells into a heart to fix the broken heart, the problem has always been, and there's beautiful data in the literature in monkeys and various other species, particularly in monkeys, that the cells do integrate, but the problem is that they generate arrhythmias. And so they generate arrhythmias and the animals die of arrhythmias. And that has been a problem that people have not been able to solve. So do you have any comments about how you would go to prevent this problem and to bring the field forward in a way that would be safe for the patient? And also, I would be interested to know which kinds of cells you would like to integrate into the materials. You want to do induced pluripotent stem cell derived cardiomyocytes, embryonic stem cells, or any other type of heart cells. Thank you.
Yeah. Yeah, so thanks for that. I mean, absolutely confirm what you said. I mean, the really big problems are way ahead of us. So far what we did, so the published story is mouse cardiomyocytes that we directly prepared from the heart. And I fully agree that to get really in the future, and I'm talking about in 15 years or so, that is my guess, a functional heart muscle that really works in vivo, we have to be aware of that it's not only cardiomyocytes, right? There's also other cells. There has to be communication. There has to be a very clear communication also between the other cells that are involved. And what we try to do is actually to get selective materials so that we actually really on demand can distinguish which type of cell is located where in the tissue. That is sort of the plan, but we are at the very beginning of that. And of course, the route towards that is not working with mature cardiomyocytes, but we started now with iPS, and so human iPS cells, which was also a bit tricky. So for 2 years, we did not get that running, but now it's running well. So we can integrate the human iPS cells, and we actually can differentiate them into cardiomyocytes in 3D spider cell scaffolds. This works. These constructs, they beat. However, we need a lot of cells. So right now we work with about 5 million cells per milliliter. So that is one of the big issues. We need enough cells to get started with. Of course, beating rates are far off what we will need then in the final construct. So this is also something we have to work on, how to stimulate the cardiomyocytes, how to get them mature. So a lot of unanswered questions. I'm just a materials developer, and we work close together with our heart muscle specialist at the University of Erlangen Clinical Center, which is Felix Engel. So he is the heart muscle specialist, so coming from the medical side, and we try to provide the materials, and we try to adopt the materials, engineer new materials to get step by step into the right direction. But I'm absolutely with you that that's the big issue that probably will not be solved within the next, I would guess, 10 to 15 years.
I agree with you, and I thank you for your presentation. I think it is very stimulating, particularly for the young people who are hearing us. I think this is an interesting way to go, and I thank you.
Thank you very much.
Well, we have another question by Conchi Serrano from the Material Science Institute in Madrid. And she says, in which range is the Young modulus of silk foams? And how do you maintain the silk roll and the chitosan silk rolls together without opening?
Yeah, so thanks for that question. So the silk foams that we use currently, they are thought for skin regeneration. So this is what we do there. And therefore we need Janus moduli in the range of kilopascal. So we're talking about between 80 and 100 kilopascal Janus modulus in the foams. And they are made by salt leaching. But of course, we can produce forms in other ways, and they then have different properties. But the forms that we currently use are for skin regeneration in the kilopascal regime. Yeah, and with the silk chitosan silk roll, well, as long as it's wet, it will stay in this conformation. And since we intend to implant that into the body, it will not get dry. Yeah. If you dry it out completely, then it will get flat again. But this is not an issue because as soon as we have packed everything, we would like to implant that, and then it will not get dry anymore. So therefore, it stays in its shape. And then, of course, the endothelial cells come in, and they also make endothelial cell layers. So the other idea is that the silk supporting material, and I forgot to mention that, it will be biodegraded over between 18 to 24 months. In this rolling tubes. So it will be replaced then by actually natural tissue. That is the idea. So we just need to have this in place for a few months, and then it will be gone anyhow, and then the supportive structure comes again from the regrown natural tissue.
Sorry. Now there is another question by Miguel Vicente, so I'm going to open his microphone. I think it's open now. Now, Miguel Vicente.
Yeah. Can you hear me?
Yes.
So Thomas, thank you so much for such a fun and whimsical talk. I mean, this was actually beautiful. So we are interested in cell migration mostly. And as you know, neuroblasts actually migrate on glial cells that extends like these thin, long processes that look very much like your collagen fibers. I mean, have you checked if neuroblasts actually migrate on your fibers or they just get attached there? Thank you.
No, yeah. So thanks for the question. Well, here I've shown you the already attached structures, but we also have the setup where we have the pre-rolled tube, and there we actually have the neuroplasts coming from outside. So they actually grow along the fibers. So they can also grow along the structure. So both is possible.
Okay, great.
Okay, thank you. Now we have another question by Daniel González from CTV, and he asks, did you check if bioprinted cells establish cell-to-cell contact? This should be important to establish intercellular communication through connexins and transmission of cardiac electric potential.
Yeah, so very good question. So I've shown you the connexin connections in the 2D structures. We also right now running the same experiments with the 3D structures, but they just have to evaluate the data so far. So in 2D that works, yes. For 3D we have tested it, but it's not evaluated yet. Yeah, of course, this is very important. So the next step for having these 3D structures is that we also will apply electric patches outside. So we would like to stimulate the heart muscle cells now with electric potentials from outside. And for that, we right now generate a bioreactor. So that actually allows us to grow this piece of silk with the cardiomyocytes, and it gets then electrically stimulated during the maturation system. And right now we set up this bioreactor through traditional 3D printing and try to implement everything in a place. This is what we're currently working on. Yeah, so it's a very good idea. Yeah.
Now there is another question by Manuel Antonio Oria, and provided that silk structure is highly stable and it can be used for biomedical purposes, for how long is the silk material going to last in the organism? And in addition, if this material are degraded, is it possible that this material serves as a base for the deposition of organism amyloidogenic proteins in order to form amyloid?
Okay, very, very good questions. I start with the last question. No, it's not the surface for other amyloids, and I know what I'm talking about. Before I went into the silk business, I was working for more than 10 years in the amyloid business and in the prion business, so Mercalli disease and so on. And of course, we tested a lot of amyloids that we have here still in our drawers. So silk is very sequence specifically inducing fibril formation. And as soon as you add something else, it does not work. So from our view and from our side, it's very clear that silk is not causing, at least in vitro, and we have never seen any complication in vivo so far. The other point, and therefore I started with that, the other point is that silk inside the human body is degraded, well, depending on where you put it, yeah, and in which morphology, more or less quickly, yeah. So we will not have any silk depots that stay there for years. It might cause trouble later, but the silk is degraded. So now it depends where you put it. And one important thing to know is that spiders recycle their webs by eating that. So if you have digestive enzymes, silk is degraded within minutes. And we tested that. So we tested stomach fluids and intestinal fluids. In stomach fluids, acidic pH, the silk is stable for weeks. In intestinal fluids with pancreatin, it's digested within 5 minutes. Then, of course, for us, more important, because we don't want to eat this silk. I mean, you could do that, but, well, not necessarily. We are more interested in wound healing devices. So, of course, we investigated how our wound proteases interacting with silk. And there it really depends on the morphology. Particles are very rapidly degraded. So we use particles for drug delivery. So for them, it takes about 2 to 3 days to be fully degraded. Yeah, because you have a very high surface to volume ratio, and this gives a lot of attachment sites for the proteases. If we have films, they are much more stable. They're stable over weeks and months, and with the pressed implants, because this was one of the questions, for the pressed implants, we see that after around about 9 months, the coating starts to get thinner. And after about 18 months, the coating is gone. But that's fine, because the side effect is fibrosis, which is the medical bad thing, so to say, only takes place between month 3 and month 9. So if the coating is stable more than 12 months, and that's the case, we prevent the fibrosis, and then the implant is fine, and it's well integrated into the human body. And then later, the silk will be degraded. The most stable structure is the fibers. Yeah? So they probably last, and there's study by Fritz Vollrath, so one of the pioneers in silk, spider silk studies. So they could show that it's stable for 2 or 2.5 years, but then it's also getting degraded over time. However, it really depends where you place it in the body, under the skin or really to an organ, because we found out that only very selected proteases like the MMP2, so the metalloproteases, the wound healing protease 2, in combination with elastase, can digest the silk. If you have the individual enzymes, none of them has any effect in vitro. So you need explicit mixtures of different enzymes, which you don't find everywhere in all the wounds. And this, I think, makes this situation very special.
Okay, thank you. We have, I don't know if there is any more questions. If not, well, once again, I would like to thank Professor Scheibel for his extraordinary talk and his time. And let me finish with just 2 personal remarks. The first is that I hope that we will be able to meet in person in a non too distant future. And the second is that I hope that Bayern München does a good match tonight, but hopefully not too good. So thank you, Thomas, and hope to see you soon. And now,
thank you very much. I'm looking forward to our next meeting.
Okay, so now we have a 2 minutes break, and then Gustavo Plaza will be the chairman for the second session. Thanks to you all.
Okay, Gustavo.
Hola. Okay, Maite. So let's continue with the seminars. Now it's time for the short talks. And the first talk will be by Rebeca Bocanegra. Rebeca is researcher at IMDEA-Nanocience at the Molecular Motor Manipulation Lab with Borja Ibarra. Rebeca obtained her PhD in 2011 in the molecular biology field. Since 2016, she has been working at IMDEA-Nanocience studying membrane behavior. Rebeca, if you are
Yes, I'm here.
available. Okay, so you can start now.
Yeah, thank you for the nice introduction. So I will share my screen now. Can you see it?
Yes.
Okay, I will show you today this work that we have recently published about the dynamic constriction and fission of endoplasmic reticulum membranes by reticulon. Well, as you all know, the endoplasmic reticulum is a continuous membrane system inside the cell, which is highly dynamic and is constantly changing in the cell cycle, and has some parts with highly branched tubules and parts with flat sheets, and it has interactions with cytoskeleton, plasma membrane, and other organelles. And the maintenance of this system is related in one hand to atlastin proteins, which are fusogenic, and on the other hand has been related to reticulon. Reticulons is a family of proteins which share a common structural feature, which is a hydrophobic part, which is able to form a wedge-shaped hairpin that spans on a monolayer or the whole bilayer of the endoplasmic reticulum and can interact with the cytoskeleton. So among the evidences that say that reticulon is involved in the endoplasmic reticulum maintenance are those from the group of Arturo Dagas, who is one of our collaborators, that have seen in animal model in Drosophila, which only has one atlastin gene and one reticulon gene, that if you avoid the expression of reticulon, you obtain this endoplasmic reticulum, which is without any branches, and with an overexpression of reticulon, the endoplasmic reticulum, I don't know if you can see here, this red dots, it's highly disconnected. They have expressed in COS7 cell this reticulum 1 gene, and in this case, they have seen the disconnection of the endoplasmic reticulum branches, and they have been able to observe the fission of some of the branches of the ER, and this fission is always occur in the zones of the ER that has been remodeled in active remodeling, of course, and this retraction of the branches to the ER is slower when with this overexpression. And the goal of our investigation in this part is to see with our technique, which is optical tweezers, we wanted to test the fission activity of reticulon and characterize the mechanics of this activity. For this, as I have said, we use the optical tweezers technique. We have two dedicated mini tweezers machines, which consist in two counterpropagating laser beams focused in the same point to generate a single trap. This is all inside a microfluidics chamber with a pipette, so we can attach our biological system of interest between one bead, which is held by suction in top of this micropipette, and another bead which is in the optical trap. This optical trap, we can move it up and down, so we can exert force in our biological system, and we can measure the forces too. So to study the membranes, what we do is to generate, I will pass it fast, generate some vesicles on top of these plastic beads, and generate between the two beads a lipidic nanotube. How we do this is, for example, with a pure lipidic sample, what we do is to put together the two beads and pull the two beads apart. This generates a nanotube, and we can follow the formation of the tube by the characteristic Forks extension curve that is recorded by the machine. In a pure lipidic sample, once you obtain this nanotube, the force that we measure is constant and is reversible and is related with the radius of this tube. In the case of the reticulon lipid sample, we have the reticulon. It's a proteolipid sample, sorry. So we do exactly the same, but the force extension curve that we obtain is quite different from the pure lipidic sample. We see this increasing force as we stretch the tube, and as we put closer together the two beads again, we see a huge hysteresis. So we have done this kind of experiments with the pure lipid samples and reticular lipid samples at different pooling rates. And what we observe is that we can do a lot of different consecutive force extension cycles with the lipidic samples, but with the reticulum samples, at some points these tubes break. Always when the tube is in movement. Of course, as we are stretching the reticulum tubes, we reach higher forces than in the lipidic samples because of this kind of behavior. But even if we pull with really high forces the lipidic tubes, we never see fission in the lipidic tube. Sorry. So here I show you the increasing force that we obtain in the reticulum tubes in the moment of fission, which is the black dots, and the maximum forces measured in the lipid tubes that I repeat that never get broken. So, and the forces are quite similar in the higher speed range. So the high forces that we obtain are not the only reason that gets the tube broken. So we can relate this increasing force in the case of the reticulum with reduction in the radius of the tube, because I have said, the forces are related with the radius. In fluorescence experiments that were performed in Bilbao by our other collaborators, Vadim Frolov lab at Iker Basque, they have seen that in static tubes, the tubes that they obtain in the presence of reticulum have these constricted and bulged parts. So, of course, reticulum is producing a constriction of the tube, as we have observed in the force traces. Interestingly, in these static tubes, the reticulum is stabilizing, is counterintuitive, but the reticulum is stabilizing these constricted parts. And this we know because if you just I don't know how to say that. If you just stop pulling the tube, or if you put together the two bits in our experiments, for example, you see this huge hysteresis and this huge decrease in the force. So reticulum somehow is stabilizing these tubes. So we have these two different behaviors here. In one hand, reticulum is constricting tubes, but is stabilizing the tube. This is related with the observations that in vivo, is mostly you can find reticulum mostly in the nanotubes of the ER and in the junction of the branches. The other observation that we have made in fluorescence experiments is that the proximal part to the reservoir, to the membrane reservoir, the tubes are even thinner, more constricted than in the distal part that is down here. From images, from fluorescence images of the tubes, and the forces that we obtain in the same samples and at the same forces and pull speeds, we have calculated the bending modulus of the tube itself, and this bending modulus of the tube is lower than the bending modulus of the membrane, the bulk membrane. This is, in our model, this is produced by the sorting of reticulum from the membrane reservoir to the tube, which is producing a viscous drag. If we represent the radius of the tube obtained from the forces in our optical tweezers experiments in front of the velocity, with the bending modulus, we use the bending modulus of the bulk membrane, we obtain radius that are, well, these two points in black are at higher velocities. This radius is below the minimum limit for the fission of the tube, but at low forces that are more comparable with the physiological ones that the cytoskeleton can exert. This radius is higher than the limit, but if we use this new bending modulus that we obtain from the thinner part, we can see that the nanotube radius is below this hemifission limit. So the conclusion is that the radius, the reticulum, is constricting dynamically the nanotube. So in conclusion, what we have observed is that reticulon 1 is using two synergistic modes of membrane curvature, static and dynamic. And the sensitivity to the membrane movement suggests that this dynamic regulation of the topology of the endoplasmic reticulum that links fusion and fission with membrane motility. So, and in the cell, this motility is driven by the cytoskeleton, and that's why reticulum is, that's the mechanism that reticulum is using to promote fission of the ER. So finally, I just want to thank all people in our lab at IMDEA-Nanocience and our collaborators in Bilbao and in Italy. And thank you all for your kind attention. I think I have talked too much, too fast maybe, but I hope you have more or less follow me.
Yes, thank you, Rebecca, for the nice talk. Virtual applause. So the plan is to have these 6 talks, and later, when the 6th talk is finished at 1:20, we will have time for questions and for discussion. Anyway, if there is a quick question, we can have 1 minute to answer it now, Rebecca.
Mm-hmm.
And otherwise, we can continue with the short talks, and then at 1:20 we will have the time for questions. I think, Daniel, you have a question, right?
No.
Ah, okay. Okay, so well, thank you again, Rebecca, and let's continue with the next talk by Daniel García. So, okay. So Daniel, he's researcher at Carlos III University at the Department of Continuum Mechanics and Structural Analysis. Daniel, in fact, he has developed his career at the University of Carlos III and at the University of Oxford. His research focuses on continuum media formulations combining mechanics and other physics. And now he has just received a starting grant. And well, so Daniel, you can start now.
Okay, so thank you very much for the nice introduction. So let me share the screen. Okay. Okay, so I guess you can see it now, right?
Yes, perfect.
Okay, so good morning, everybody. Well, as you say, I'm much more modeling guy. So what I'm going to present now is some modeling approaches in biomechanics. So we'll present a very recent work on collective cell migration, then move to briefly introduce some brain mechanics. So regarding motivation, when we look at different biomechanical processes, such as cell migration, traumatic brain injury, so these two ones are going the ones I'm going to provide more details, but those involve healing or cardiac function, especially the last one. We have this electric response strongly coupled to the mechanical pumping of the heart. We realize that mechanics plays a major role. So when we look inside these mechanics, what we realize this is far from being simple, and we have too many different dependencies. So we can have some temperature dependence that modulates the stiffness of the biological tissue. Also, we can see that these biological tissues are not purely elastic, right? So we have some viscous contributions. And regarding these viscous contributions, we have some consequences, such as here, strain rate dependency, so that depending on the rate we apply the mechanical load, we're going to get usually different stiffer response, right? So the higher the strain rate, the higher the stiffer the mechanical response of the system. And this is also intrinsically coupled to this kind of viscous relaxation, so that if we apply a given load at a given rate and then we keep that load, we can observe kind of a stress relaxation with time. And on top of that, we have many other different dependencies on top, just to mention one, so anisotropy in muscles or tendon tissues, where we have some fibers, and we can see that we have different mechanical response if we apply the load in that longitudinal direction or in the transverse. So something common to all these dependencies is that in the end, so this is not just Young's modulus, right? So this is not just some linear viscoelasticity. So we have strong nonlinearities, and this is important not only from the modeling perspective, but also if we are analyzing some experimental characterizations, we can see that, well, not exactly this, but in the true stress to strain, so this stiffness is actually the slope of this curve. So if we are reaching enough large deformation, everything is changing and we can have even order of magnitudes of difference. So now I would like to introduce some works where we are playing with these mechanics in a kind of modeling framework. So this first one actually is in collaboration with Arrate from Universidad Carlos III of Madrid as well. So we have recently published that in Stream Mechanics Letters, and to be honest, this is quite new topic for me. So really happy to have any comments or feedback from you guys. So when we look at cell migration, from an individual point of view, we know that these individual cells have some protrusions, right? And some inactive stresses with some contraction or expansion. And this is far from being kind of isolated system. We have some interactions with the extracellular matrix, substrate, or even with other cells. So in the end, we have some interaction forces and some internal forces, really. And as soon as we have some links between different cells, all these mechanical forces are going to be transmitted somehow through the whole system. So just to mention two examples of collective cell migration, we have here cancer cell invasion. So this few have to say, I thanks Dennis Beer from Johns Hopkins University to provide here, because we can see how we follow the time event, we can see kind of continuum-like expansion of this kind of collective cell migration. And I think this is quite nice here in this video kindly provided by Xavier Trepal from Barcelona, that we can see how these forces from individual perspective are introduced some forces that are somehow transmitted to the whole event. And something I would like to highlight here is that we can see that we have regions with large deformation. So in this point, we are losing this kind of linear elasticity, and we need to move to other hyperelastic theories. So looking inside this problem, more from a theoretical perspective, we need to identify different governing equations. So I just mentioned here three, but actually we'll focus on these two latter ones. Just mention we can imagine we can have some proliferation dynamics or other chemical diffusion-like processes actually we're incorporating now. So one fact is that we can consider this concentration of contractile actomyosin. So in the end, we're going to have kind of active stress, right, related to that contractile actomyosin, or we're going to have some contraction or expansion. Then we jump to this cell polarization dynamics. So what this is telling us, okay, so your cellular system can change in shape and react, right? So in the end, all these forces are going to have any kind of directed resultant. And then in the end, we have this mechanical balance that to me is the most important one. This is kind of cocktail where we can put all the different forces together, and then somehow this is going to determine the fate of this collective migration. So having this in mind, we wanted to focus on a very specific problem. And actually we took this experimental data published by Sanjay et al. In Science. So basically we have some epithelial monolayers cultured in different substrates. So we can observe here in the left-hand side image that when we have kind of uniform substrate, we can observe kind of collective cell migration in a very symmetric fashion. But once we move to an heterogeneous substrate, we have kind of a stiffness gradient, right? We can observe that this migration is directed towards this stiffer region. So when we look into the literature, most of the experimental works and even some, nice modeling frameworks suggest that this is somehow a kind of stiffness-dependent propulsion force, so that as our system is somehow connected by focal adhesions to the substrate, we can expect higher interaction forces here in that stiffer region than in the softer one, so that we're going to have kind of resultant force directing the migration to that region. But if we look at the, some experimental works in the literature also, we realize that this is not only this propulsion force, but also somehow of a stiffness-dependent polarization. So we can observe that this cellular polarization depends on both the magnitude of the substrate, but also on some stiffness gradients. So at this stage, we wanted actually to provide kind of simple modeling framework to try to test these different hypotheses and determine from a mechanical point of view what is the implication of each one. So we started with the mechanical balance. So we provide here somehow together these internal forces and external forces. So we will have this internal cellular stress ponderated by the height of the epithelial monolayer. So here sigma is the cellular stress, and T is the traction forces. So here we say, okay, we're going to have this kind of propulsion force that a priori can be dependent on this stiffness of the substrate following these previous approaches. And then the direction of that resultant force is going to be somehow determined by this cellular polarization. And then, of course, as soon as we have any kind of system moving on top of the substrate, we're going to have some friction forces, right? Opposing to that with this parameter being this friction coefficient. So once we have defined that, we need to define what is happening inside the cellular stress, right? So basically, we said, okay, let's try to use some hyperelastic theory. So the main idea here is that instead of defining directly the constitutive relation, we're going to define a kind of mechanical potential. So basically, this is the mechanical energy, right, that is stored in the system and we can recover at some point. So using some solid mechanics and thermodynamic principles, we can reach the following consistent relationship between the deformation state of the cellular system, defined by this F, that this is the deformation gradient, and our Cauchy stress tensors, our true stress. And then we can observe here that we have kind of two components, with this first one being a volumetric one, with this P being kind of pressure that we are going to get from boundary conditions and some consistency equations. And then we have here a second term related to the shear, where this mu cell is the stiffness of the cellular system. So basically the shear modulus. So once we have defined that, let me come back here and we say, okay, so these traction forces are going to somehow depend on how this cellular polarization is. And we can expect this is going to somehow change during the whole event. So we decided to provide this kind of governing equation for the cellular polarization. So we are aware there are two other many terms we can include, like dependence with velocity or others. But we decided to provide these two ones to test the different contribution of our hypothesis. So first, this blue term is saying, okay, so because of this stiffness gradient, we can have some cells that are going to polarize to a stiffer region, right? So then we have this first term that is saying, okay, as all these cells are somehow linked and add each other, we're going to have a kind of diffusion-like process where all these other cells are going to lead to that polarization. So then, even if we keep kind of constant propulsion force, what we're going to have is kind of resultant force, okay, towards this stiffer region that is going to be able to capture this kind of tendency in the migration dynamics. So just at this stage, I'm going to skip all the mathematics. Basically, what we did is implement that in a finite element code for some implicit integration algorithms. So that say, we're solving all the different equations at every single spatial point at the same time, finding for the exact solution for the problem. So once we have everything implemented, we were happy with that. We say, okay, so let's try now. Let's try to validate the model. Let's try to evaluate the predictive capability. So we made use of this experimental data that I mentioned before from Sanjay et al., where we have kind of evolution of this migration dynamics on top of homogeneous substrate. Okay, so no stiffness gradient at this stage. So at this stage, we did no fitting. So basically, we took some parameters directly from the physical meaning and from some experimental evidences. And what we can observe first in this panel A is that in black dots, we have the evolution of the experimental tendencies. And in red, we have the evolution of the model prediction. And this is basically distance to midline along time. So how this monolayer is expanding along time. So we can see a very good agreement by means of these migration dynamics between experiments and model. But we say, okay, so we're happy with that, but we need some extra validation, some double check. So what is happening by means of mechanics? And then we compare experimental results with some models by means of stress traction. So we can see a very good agreement. Naturally, if we come here to panel B, we can see that the maximum stress traction from an experiment is almost identical to the one predicted to the model. So at this stage, we were happy with that. And we moved to test the model in heterogeneous substrate. So at this stage, we split all these simulations into main sets. So this first one, all these red lines are related to some stiffness-dependent polarization. So what we're going to say here, okay, so let's consider this propulsion force is somehow constant. And then everything is coming from this cellular polarization following this kind of gradient stiffness. And then in the other way around, so for the blue terms, we have that we skip this bit and we just focus on a propulsion force depending on the stiffness. So what we can see here is that by means of migration dynamics, we have almost same tendencies. So we can say that at this point, both hypotheses can explain this kind of migration system. But then we look at the, the result by means of mechanics. So let me go to the following slide, but this is, I think, more clear here. So what we can see is that this stiffness-dependent polarization, we can predict this kind of migration toward the stiffer region. And if you realize we're keeping the symmetric distribution of the stress, and then we're just introducing kind of a slight increase in stress. But if we go for this kind of stiffness-dependent propulsion force, what it implies is that we are losing this symmetry, and also it means that we're going to get kind of much higher internal stress. So I just wanted to highlight the usefulness of this kind of modeling approaches to complement experiments and to test hypotheses, right? Because at some point we have the experiments with some observation, we launch some testing hypotheses, but we don't really know which are the consequences by means of, for example, mechanics in this case. So at this point, let me jump to what I call private story and brain mechanics. So just want to make highlight a couple of points here. So just going to present some previous works I did actually during my postdoc at Oxford with Professor Jerusalem. So basically here, we were focused on central nervous system injuries. So that, okay, we have impact on our head during sport-related activities, fall of a person, or even in a military context. We know there are some consequences by means of pathology, brain dysfunction, or even at the cellular level, we have some alteration in the action potential propagation, right? So at this point, we know there's some biomarkers such as oxidative stress or others, but we thought, okay, so of course, one cause of that is that we have kind of mechanical deformation of the brain tissue itself. So there should be some mechanical variables governing all these CNS injuries. So in the end, we wanted kind of threshold saying, okay, so at this threshold, we can expect some injuries, below that not. So we were doing some collaborative work with people from Purdue University, and we realized that from an experimental point of view, it's almost impossible to track the whole event, looking what is happening inside. So we decided to go for computational modeling. And what I want to highlight here is that once we have some simple models, like the one I presented before, once we're happy with that validation, some calibration, we can jump and go a step beyond, right? And we can now incorporate these models. Obviously, we will need extra validations for that, but we can incorporate that in more complex geometries, in more complex loading and boundary scenarios. And in that case, for example, we were able to see that in a closed-gate impact, we can have this kind of distribution of the stress and deformation within the brain, while in blast explosion, we can follow the whole mechanical event and see that the patterns of these mechanical distributions is completely different. So just wanted to close. It's not just conclusions, I would like to mention some ongoing work and future work. So basically, my conclusion here is that obviously mechanical environment, it's playing a major role. So my point now is, okay, let's try to control the cellular mechanical environment. Let's try to control this remotely. So what I want to propose here is to use some magnetoactive polymers, magnetoactive hydrogels really, to try to control the different mechanical properties or even mechanical deformations by the application of an external field. So the point here is that this is quite complex problem. And of course, we will need experiments, theory, and modeling. So yeah, this is what I'm really focused now. Some magnetoactive hydrogels where we're going to have some mechanics that are interplaying with different magnetic fields, right? So applied magnetic field can introduce some mechanical changes. But of course, as we have this hydrogel nature, this is not a viscoelasticity just. So we will have also some solvent diffusion, some proelasticity. So my idea here is, okay, let's try to build up kind of consistent computational framework, so we can understand the physics, we can design properly this kind of substrate, and let's try to control the cellular mechanical environment. So I think I ran out of time, so don't know if there is question. If not, I'm very happy to have some contacts, look for collaboration. And just to mention, this is what this ERC starting grant is about. And well, happy to get feedback, comments, whatever you consider. Thank you very much.
Thank you, Daniel. Thank you for the nice talk, presenting your research. So yes, we have 1 or 2 minutes if there is a question now. Otherwise, we will have time, as I said, at 1:20.
Nice.
So, well, I think it's good to be on time, you know? So we can continue. Thank you again, Daniel.
Thank you very much.
And we can now go to the next talk by María Rosaria Bricolo. Hi, Maria. She's a researcher at CENIC, at the group of molecular mechanics of the cardiovascular system with Jorge Alegre. She obtained her PhD in molecular medicine and medical biotechnology in 2019, and then she joined this group at CENIC. María Rosaria, if you are ready, you can start now.
Yeah, I'm sharing my presentation. Okay. Can you see the presentation?
Yes, perfect.
Okay, so fine. First of all, thank you to give me the opportunity to be here today. Let me start. I'm sorry. Okay. Let me start my presentation with a brief introduction about the mechanism of cell mechanosensing and mechanotransduction. As you probably know, cells sense the externally force, like, for example, the movement or gravity, but also sense the external internal active force, like, for example, induced by the changes in the cell contraction or shape. And they transduce this mechanical signal in a more simple language for cell, which is written in biochemical and metabolic words. We have a long list of force-bearing proteins, including, for example, tallin, which is important in the coupling between integrins and actin of cytoskeleton, but also, for example, we have lamins, important for the maintenance of mechanical integrity of nucleus, alpha-catenin with in the cell adhesion, and also titin. Titin has a K rule in the mechanosensing, mechanotransduction in muscle cells. This generation and sensing of mechanical signal determine the cell and tissue behavior in heart and disease. And the heart, for example, responds to mechanical cues with remodeling processes, tissue remodeling processes. And in these processes, they play a key role, the connection between cardiomyocyte and fibroblast. However, when we have, for example, the pressure overload in the heart, we have a tissue remodeling and such response like hypertrophy. When we maintain this mechanical overload, we can have the shift of the homeostasis of the cardiovascular system toward maladaptive remodeling of the tissue. As I say before, titin play a key role in the mechanical sensing and transduction cardiomyocytes. Titin has an important role to the maintenance of passive stiffness in cardiomyocytes, and has this mechanically active region in the A band, which is a spring-like region in which we have changing in the in the conformation of this region. Yes, for example, during the contraction relaxation of myocytes, we have the uncoupling or recoupling of the this uncoiled region, or the unfolding and refolding of immunoglobulin domain of the I band. So essentially, these conformational changes can alter the binding affinity for interactor. So in general, for the first binding protein, we have an active sites, and also we have accretive sites, which is a response to changing in forces. So for example, at low forces, this cryptic sites is buried in the structure, and so the site is off. But during the stretch of the protein, we have the unfolding, so the conformational changes, and the so the high force induces the exposure of the cryptic sites, and which is result active. Now, several approaches have been implemented to examine the protein mechanics. Like, for example, we can use, for example, the traditional knockout or knockdown, or the overexpression of protein in cell. But in this case, we can alter the mechanosensing part of the protein, but also no mechanical function part of the protein. So the question is how we can study the role of protein mechanics in physiology and also in vivo, so in living matter. So our proposal in our project, we want to start the implement of the tool in which we can analyze and modulate the mechanical property in living cells and animals. And we decide to design these two models using Titin, which has mechanical property, and we call these two models MLOF, so mechanical loss of function and mechanical gain of function. In the mechanical loss of function is based on cleavage of the here, the cleavage of the first transducing protein by TEF protease. So we have essentially a sequence for the recognition of TEF protease, and this cleavage locks the sites in the state off. Whereas in the mechanical gain of function, we using is achieved by the SPI-TAG SPI-catcher system, in which we have the clamping of the random coil region, which leads the high force experienced by the domain. So hence we have in this high force manner the site is exposure and is maintained to the active states. Thanks to our collaboration with Julio Fernandez of the Columbia University and Wolfgang Linke in Münster, my collaborators, my colleagues, developed this mice model, the HaloTag TEF Titin mice model, in which we have the TEF site in Titin, the I band of Titin, and also in the safe cassette we have this protein, the HaloTag protein, which allow us the labeling of Titin in vivo. So they found that the insertion of the cassette is well tolerated in mice, and thanks of the HaloTag, we can see the structure of the sarcomere because we follow the Titin in vivo, and also they found that in this insertion is maintaining, not perturb the structure of sarcomere. The ex vivo and in vitro experiment demonstrate that TEV protease mediate the severing of the titin, in which we have the TEV site of titin. So thanks to this model, we can follow in the protein in vivo, and our idea is to use this model also to following the severing of titin in vivo. So essentially, we want to check the effect of the MLOF and NGOF model in cardiomyocytes, but also during the differentiation of cardiomyocytes and in vivo model. So to do that, we need an inducible expression of our mechanical effector represented by TEV, of course, in the MLOF model, and also the spike catcher for the MGOF model. So to do that, first of all, we isolated mouse embryonic stem cell from halotitin mice, and we demonstrated that we can differentiate this line in cardiomyocytes, and also we can follow, we can see the cardiomyocyte differentiated thanks to the halotag in titin. So our idea is to use this cell line to generate a cell line in which we can induce the expression. We have the structure for the TEV protein, and we can induce the expression of TEV in this model. So first of all, we building a lentivirus vector, in which we have, of course, the inducible promoter and TEV linked to the cherry tag, so a red fluorescence protein. As you can see here, the transfection infection using this vector, this strategy work well in HEC293 because we can see the fluorescence, so the positive fluorescence for cherry, but we didn't see the protein in Western blot analysis, so we can move it on. We can move it on another construct in which we have the same promoter, so a lentivirus vector, but without the fluorescence tag. And as you can see here, after the induction, cell express TEV after transfection and also infection with the virus. So the system work well with this vector. But an alternative tools that we can use to express TEV is also the using of adenovirus vector to express the mechanical effector. So for that, we tested another vector in which we have the cherry TEV protein in the adenovirus serotype 9 vector. And this vector works well in the HEC293. So we decided to move on the neonatal cardiomyocytes. So we infected this model of neonatal cardiomyocytes in which we have the positive fluorescence, the green fluorescence for the cardiomyocytes. And so we are able to see the expression of these cardiomyocytes, as you can see with the arrow, so the expression of TEV in these cardiomyocytes. So we decided to infect also the neonatal cardiomyocytes derived from the halotitin mice, and we found that we have the expression of cherry TEV after almost 10 days after infection. So we can say that we are able to express TEV in this cells, and also we found that the cell after infection, so after TEV expression, still remain alive, and apparently we have the the beating of this cardiomyocyte is maintaining. However, when we analyze the contractility of these cardiomyocytes, we found that we have increasing of duration of contraction after TEV expression. So we can say that, of course, we have our MLOF in a model, and this model works well. And I can say also that we start to say an altered cardiomyocyte contractility induced by a cleavage of titin. In the other hand, we also started with the building of our MGOF model, and we validate this model in vitro, in which we test our recombinant target represented by our random coil region of titin with this two hand with the spike tag protein, and the recombinant effector represented by a bifunctional spike catcher with halo. So as you can see in the SDS page electrophoresis, we have the shift of our target in presence of our effectors. So essentially, we have the coupling of our model in our model. So we can conclude that, of course, we are on track. So there are preliminary results, but we can say that the halo tag make possible the following the protein in vivo. And I guess we can say also that the severing of titin makes possible the alteration of its mechanical function. And also it's important to follow, for example, the serial titin in the differentiation of cardiomyocytes. We need an inducible expression. So in the next step, we want to produce another mice line, which we want to introduce the cherry tab in ROSA 26 locus. And also we want to check if using another serotype of adenovirus, we have a more efficient infection of cardiomyocyte, neonatal cardiomyocyte. And finally, about the MGOF, we want to, of course, start the genetic engineer to introduce the MGOF model in mice. Let me thank the molecular mechanics of cardiovascular system group at CNIC, and especially Diana, Carla, Angel, and Manuel of my group, the core facility, our funding, and of course, thank you all of you for the attention.
Thank you, Maria Rosaria, for the nice talk. So let's move to the next talk by Blanca González. Blanca is doing her PhD with us at the Center for Biomedical Technology of the Universidad Politécnica de Madrid. She studied materials engineering and industrial mathematics, and now she's working on cell mechanics and cell biophysics. So Blanca, if you are ready, you can start now.
Yes, thank you. Okay, so hi all, it's Blanca up here. I would like to say thank you for the opportunity to talk today, since it's my last year of PhD, and well, I think it's a good training to learn how to compress data within few minutes for the thesis defense. So the topic of today's presentation actually represents one of the major projects that we have developed through my PhD thesis, in which we try to evaluate the contributions of immune cells' biomechanical features in immune dysfunction and aging. Okay, so first of all, when we talk about age, for example, we typically use the concept of chronological age. So in my case, I would say, my chronological age is 28. And it is true that biological malfunction and increased risks of pathologies come with this chronological age. However, the term chronological age is also misleading because there is remarkable variation in aging rates and expected lifespans within individuals of the same chronological age. So instead of the concept of chronological age, there is this other concept, biological age, which is rather used as a better indicator of health condition, remaining healthy lifespan, and life expectancy. So biological aging is usually explained, probably you all know this diagram, as the byproduct of molecular and cellular changes that can be considered as hallmarks of biological aging. So rather than reading this whole list of hallmarks, because any one of these could cover several presentations, I just introduced the one related to this work, which is immunosenescence. So immunosenescence refers to the gradual deterioration of the immune system brought on by the natural aging process. Immunosenescence involves both the host capacity to respond to infection in a worse way, and also the development of long-term immune memory, especially by vaccination, which also deteriorates as we age. We can see in the diagram, for example, how when we become elderly, we have lower levels of naive T cells. We have a larger number of senescent T cells, and we also have larger numbers of inflationary T cells. Okay, so this age-associated immune deficiency or immunosenescence happens to everybody, both in long and short-lived species, such as us, Homo sapiens. And even in species that only live 2 years, we can also see this process as they age. One of the interesting features about immunosenescence is that besides the gradual decline in both the adaptive and the built-in immune function, it appears the process called inflammaging. Inflammaging is the chronic low-level inflammation. It is the underpinning, well, inflammation is the underpinning cause of most of chronic diseases of aging. So if we want to treat our bodies in a way that we won't have any of the diseases associated with aging, such as diabetes, cholesterol, heart diseases, or arthritis, the common thread of all of them is inflammation. Well, somehow also it is interesting to research immunosenescence as a direct cause associated to this inflammaging process. So the motivation of our work enters in this part of the presentation because as biophysicists, we also wanted to include in the frame of immunosenescence the biophysical features of cells, in the sense that there are several relations between cell states, right, and biophysics of the cells. For example, very clearly in the case of immune T cells, it's the function of T cell migration. Here in the diagram, we have a leukocyte extravasating blood vessel. And for this particular function, the cell has to deform through the interstices. And for this, a proper set of mechanical features is required. Now, in the vivo video that you can all see in the presentation, well, we have this permanent migration of T cells in red, moving through endothelial tissues. So the aim of our project was to evaluate the biophysics scenario in the age-associated changes of immune T cells. How did we do this? Well, we designed a longitudinal experiment study using as animal model mice, and we performed several characterization. We performed several characterization techniques at 4 age groups. So first of all was the young adult groups at age 40, around 40 weeks. Then at the mature age, when the individuals were 56. Then at the old age, at 72 weeks. And then when they were very old, around 96 weeks. This was in collaboration with the group of De La Fuente at Universidad Complutense de Madrid. So then for each time point that I just showed, we performed the following experiments. So the cells were extracted from the peritoneum of mice, And then the global part of the global populations of these cells were placed in Boyden chambers because we wanted to see how functional, in the sense of how these cells were able to spontaneously migrate through pores. Then with the remaining global populations of these cells, we sorted our cells in four cell populations. We investigated CD4, CD8, both of them in naive and memory states. Once the cells were sorted, we performed aspiration tests using micropipette aspiration. Then with part of the sorted cells, we performed fluorescent immunostaining, and half of the cells were set in glass slides so that we could see the morphology of them, and half of them were put in cytometry tubes so that we can analyze some proteins of interest traditionally related to the migration and mechanical properties of these cells. So we had, like, in the end of each time point, these four aspects of the biophysical landscape of T cells. Then, yes, for you to have an overview of the type of experiments we perform and the data we obtain regarding the functional assay, you can see in the images how the Boyden chamber is covered of micropores of approx the same size of the cells. And when we move up with the microscope, we can see in the lower part of the chamber those cells that have been able to migrate through the pores. So in this way, we can, for example, assess the density of cells per experiment that have been able to migrate through the pores. Regarding the deformability, this is a typical example of a micropipette aspiration experiment. We see the whole cell being aspirated as we decrease the pressure, the differential pressure inside the pipette, and the type of graphs that we obtain are automatically processed, and we can obtain parameters such as the elastic modulus, the apparent elastic modulus, the apparent viscosity, and the variation, the coefficient of variation of these parameters per experiment. Regarding the internal composition experiments, we are provided with the information of the flow cytometry in a way that you can see here. We can represent, for example, protein of interest, expression of protein of interest versus forward scatter light or whatever other parameter, and different parameters or features of the proteins of interest that we were investigating can be in this way quantified. For instance, we looked at actin, myosin, bimentin, 5-methylcytosine, which is associated with the condensation of the DNA itself, cell fluorescence by lipofuscin molecule, and the relative frequency of sorted cells, so that we can check whether there are more CD4, CD8, or naive or memory state. And finally, regarding the morphology experiments, well, in the confocal, we get images such as the one that you can see by performing thresholding, segmentation, and if we want to measure, for example, relative size of the nucleus, we perform an ellipsoid fitting, or if we want to see the actin cortex, we perform other type of Gaussian fit through the cortex. And overall, we have a set of morphological indicators of actin, DNA, and lamin B1. So now, once we have all the experiments performed, after the longitudinal study finished, we move to the analysis of the data, first by the basic quantification of the biophysical parameters that I just explained in the first type of characterization experiments. Then we wanted to see how these parameters were correlated with age. So we performed a correlation analysis. Then we also wanted to investigate whether there were more or less similarity between, for example, T cell types, CD4, CD8, memory naive, or parameter class. We are talking about mechanical parameters, morphological parameters, etc. And finally, we also wanted to investigate whether there were more or less similarity between individuals, or if this difference was not existent. So regarding the statistical tools that we use to investigate the correlation with age, well, we had the parameter listed according to their class, and we performed this kind of Spearman correlation test through which, well, using a regression model, we can estimate the extent to which the correlation, the linear correlation between the parameters and age is strong, moderate, inexistent. And the typical way, as you know, for doing this, for example, is to set a threshold value of P. And in this way, we can see whether there are some parameters that decrease with age, or some others that increase with age, for example. And then for the similarity analysis, the statistical tool that we have used for this purpose was the Jacquard index. So what we did was to represent the set of parameters, as you can see in the radar plot. So we have in each color age group, so young, mature, old, or very old. And then in each vertices of the polygon, what we have is one parameter normalized from 0 to 1. So the Jacquard index, what allows us is to quantify the intersection between the different polygons. So the ratio of the Jacquard index is given by the intersection of two sets divided by the union of the two sets. So 0 Jacquard index means that there is no intersection. 1 Jacquard index means that there is a complete overlap of the two sets. So in this way, basically, we can also create a heat map by investigating the Jacquard indexes of the different polygons that we are able to draw. So the first kind of, well, I'm going to show the raw data or the raw results obtained for the first step, which was to investigate how the parameters correlate with age. And well, we can see, let's say qualitatively first, some kind of conclusions such as that there is a significant age-related increase in mechanical parameters. For example, we observe in the 4 cell types that we investigated as cell stiffening, as cells age. We also found out that there are also changes in the sorting relative frequencies. For example, the ratio CD4 to CD8 decreases with age, and the ratio of memory to naive cells increase with age, as expected in this way. Also, now regarding the internal ordering given by the protein levels, we, for example, found that lemmin B1 decreased with age, that also 5-methylcytosine decreases with age, and that cell fluorescence of cells increase with age. And finally, regarding the functionality, we also appreciated a loss of spontaneous migration in our experiments. Regarding the similarity analysis, well, we managed to display all the data, as I just commented, to evaluate the Jacquard indices of similarities for each T cell group. No? So we draw this kind of plots for CD4, CD8, memory, and naive state, and we compare polygon versus polygon in order to have a Jacquard index. So the results are here and need to be further processed, because we now want to move to the questions of, are there similarities, for example, between age groups? Are there similarities or difference between individuals? And this is an ongoing work, so that's why I prefer to simply give this general overview of our results, and hopefully you can all come to my thesis to see the final results. And well, so the conclusion of my presentation, as I explained in the introduction, we wanted to include the biophysical features in the landscape of the immunosenescence biological process. Well, so far, regarding, for example, mechanical features, we see differences significantly correlated, for example, of stiffening of cells with age. Regarding functionality, we also have found decrease of spontaneous migration. Regarding internal ordering, well, two examples are lamin B1 and 5-methylcytosine, and we want to correlate this protein and molecules with, for example, the loss of nuclear compactness that we also found in our morphological studies. And where regarding the similarity analysis, we for the moment found that there is less similarity among agent groups than cell types. And as I said, this is ongoing work, so in the future we want to further analyze our results to investigate in individual variation and to combine all the parameters in order to perform cluster analysis as well. And this is all. Thank you to our collaborators for sure and the funding opportunities, and to all of you for your attention. And if you have questions, I guess in 20 minutes we'll answer them.
Thank you, Blanca, for the nice talk. So again, we have some time for a quick question or a couple of questions now. Or otherwise, we can just move to the last talk, and then at 1:20 we will have time for the questions. So yes, thank you again, Blanca. And the next speaker is Ángel Merchan from the Cajal Cortical Circuits Lab, directed by Javier de Felipe. So Ángel obtained his PhD in telecommunications engineering. Then he did a postdoc at Harvard Medical School. He later joined the group of de Felipe, and now he's working at the Center for Biomedical Technology of the Universidad Politécnica de Madrid. And he has been working on the study of the structure of the cerebral cortex of the rat, mouse, and human. So, Ángel, if you are ready, you can start now. We can't hear you. I think you need to turn on your microphone.
Now you should hear me.
Yes.
Okay, I'm trying to share my here. Okay. Bueno, this should be at the beginning. It's at the end. Well, first of all, I'm not a doctor in telecommunications. I'm a doctor in medicine, okay. So I have no idea of mechanical engineering or physics, and formulae, mathematical formula, etc. A few years ago, we were interested in knowing how the movements of blood vessels in the brain would change the behavior or would interact with synapses, perivascular synapses, which are located around this vascular network. Okay, as you know, blood vessels pulsate, but we were not interested in this kind of movement. As you go to thinner and thinner ramifications of vessels, this pulsation is dampened and other kind of movement takes protagonism. And this movement is the possibility of contraction and dilatation of the blood vessel. And this is made with the aim of controlling the blood flow to different regions of the brain. As I said, our interest was to know if this vessel movement could affect the synapses that are located around the vessel wall. Since, as I said, we had no idea of how to approach this mechanical problem, we got in contact with Miguel Urrecha and Ignacio Romero, who are material engineers, and we decided to build a model on this. This image is blood vessels, and in the brain, we have removed everything around them. We have removed any neural tissue that is around these vessels, but please pay attention to the scale because these are very, very small blood vessels. As I said, these are the vessels whose movements we were interested on. Well, this should advance, but it doesn't. Okay, so we had to model the blood vessel and its surrounding tissue. And the idea was to select a region of the brain where blood vessels are characteristically very active with respect to their movements, and to simulate or to build a model of the tissue around. This is the same image as before, and just think we selected a region like this one or this one with a very small vessel across 10 microns thick, okay? And around the vessel, this larger cylinder of tissue. And we are going to simulate the movements in this very simplified system. We first needed to know how vessels move. And we went to the literature and found that, well, blood vessels react to almost anything which is around them in the brain. In this particular example, you have several kinds of inhibitory neurons with the neurotransmitters they release, and they all affect the blood vessels, either for contraction or for dilatation, okay? And don't forget glial cells. Glial cells are not neurons, but they also very closely interact with the blood vessel wall through these specialized structures that we call glial end feet. They cover the outside of the blood vessels, and they communicate and interact with the vessel wall through this specialized end foot. Okay, here, of course, you will have many other neurons. Excitatory neurons have not been represented in this particular example, but their neurotransmitters also affect the vessel movements. Okay, how these substances, how these signals affect the vessel movement? Well, they change. They evoke contractions or dilations in the vessel that evoke local changes of diameter, local changes of caliber. Here you have a couple of samples from the literature in which we can see a dilatation of the blood vessels or a local, very local contraction of the blood vessels. It depends on the stimuli that dilatation or contraction are stronger, or they spread a longer distance in the blood vessel. Okay, as you see, changes in diameter are variable in percentage. So here we have another example, different substances, different stimuli, procedures, and the diameter change in this example is much higher than in the previous one. So we assumed a compromise solution, and we made our vessel to contract or dilate to a maximum or to a 20% of its original or base caliber, base diameter. At rest, they will have 10 microns of diameter, and variation of this diameter would be plus minus 20%, okay? Next, we have to simulate the perivascular tissue in the brain with special attention to synapses. And we paid special attention to synapses because these are, as you know, the places where neurons communicate with other neurons, okay? And there, in these particular places, is where these many substances are released. These many substances that can interact with other neurons, of course, and also with the vessel wall. So synapses were our main interest when we performed this simulation of the vessel walls. To do that, we based the location of synapses was based in what we know from three-dimensional electron microscopy. Basically, three-dimensional electron microscopy is based in serial sections from which we make a reconstruction, and in this case, you're going to see how we reconstruct the places where neurons connect synapses within this volume of tissue, which is reconstructed from serial sections. This perhaps you don't recognize them as synapses because only the point of contact has been represented, and we can reduce or simplify or idealize the system even more if we consider just the centroids or the coordinates where these synapses are located. Please remember that we haven't reconstructed the whole synapse, I mean the axons, the dendrite, etc., but only the point of contact. In green, excitatory synapses, in red, inhibitory synapses. And these centroids are the positions in space of these synapses. Of course, this follows a rule, and I can tell you that synapses in the brain, at least in the cortex, in the neocortex, are located almost at random. So if you follow this model, if in a model you want to construct, you put synapses more or less at random, they will reproduce what happens in real tissue. Of course, we were interested in what happened around blood vessels. You can here see a blood vessel, and we are reconstructed the blood vessel and the perivascular synapses with the same method as before. And you will see that very similarly, as you've seen before, synapses are located everywhere, everywhere, randomly or almost randomly distributed around the blood vessel too. Okay, green synapses are excitatory, red synapses are inhibitory, much less frequent in the cortex. Well, if we put everything together, we made, I repeat, with the help of these Miguel Urrecha and Ignacio Romero, the engineers, they made this finite element model in which we have represented here half cylinder of the model, and you see here the blood vessel crossing the tissue, and we have represented here perivascular synapses with the same color code as you've seen before. In this case, please remember that we haven't represented all synapses that are located around the vessel. We have only represented 1/10 of the whole amount of synapses that surround a common blood vessel in the brain. What we are going to do is simulate a local contraction and dilation in this central region of the model, and then we will measure. I hope you can see it. I'll repeat it in again. Contraction, dilatation, contraction, and dilation, okay? And as you see, synapses around the blood vessel move too, because, as you know, the brain tissue is very, very soft, okay? We can measure in our model the maximum displacements of the surrounding synapses in the model, and you can see here how the maximum displacement is located here, okay? Right at the level where the deformation of the vessel wall occurs. And in this case, we have simulated symmetrical deformation for contraction and for dilation, okay? It's interesting to note that the deformation of the surrounding tissue is quite local. I mean, it doesn't spread to the whole model. It only takes place very close to the moving vessel wall, okay? It only takes place close to the vessel wall. And this is interesting. We will discuss later why. We can measure these displacements. And as you can see here, these are different time points, okay? If we concentrate in the maximum displacement, okay, it's 1 micron, but it decays to 0.5 microns, 5 microns from the vessel walls, okay? So whatever happens at the vessel wall, it decays to 1/2, 5 microns far from the vessel walls. And it decays again to another half, 12, 15 microns farther, and so on. So the closest you are from the wall, the most sensitive to these movements are the positions of synapses. This can be represented also by the distance increment between synapses. This is important because if synapses are far from each other, the neurotransmitter that they release always spills over around in the vicinity, in the neighborhood of the active synapse. But if they are close to other synapses, they could crosstalk. I mean, this is a phenomenon that is very interesting because in this case, synapses would not act as individual contacts, but somehow would interact between them, and they act as a collective entity, okay? So if synapses are far enough and we compress them and they close them and we put them close enough to crosstalk, we are changing the way the cortex is behaving, okay? But again, this phenomenon is only restricted, let's say, to 5 microns surrounding the vessel wall. So this can be represented as a field of displacements. And as you see, imagine the blood vessel is here, okay? The vessel wall is here. Remember, only this part of the vessel wall will move in response to a contraction or dilatation stimuli, and this is the tissue surrounding the vessel wall. As you see, the deformation of the perivascular tissue, and of course the changes in distance between synapses is restricted to a very small region around the vessel wall. Okay. We can represent this dynamically with the same color code. You see synapses moving in this region, but not here, here, or here. Okay. Contraction, dilatation, and the perivascular tissue is only affected locally. So, in conclusion, blood vessel movement change the position of cyanation close to the vessel wall. Okay, this movement causes local changes in the density of perivascular synapses. The density increases when the vessel dilates because it compresses the tissue around it, and the density of synapses decreases when the vessel contracts because it expands the tissue around it. Okay, these changes may help to spread or restrict the diffusion of perivascular released signals. Okay, as I said, if sinuses are forced to stay together, more closely located than before, the neurotransmitter that is spilled over around them will crosstalk with other synapses, and also will be able to interact directly with the vessel wall. Okay, so here we have a possible mechanism of interaction or regulation of the vascular activity. Okay, and of course, don't forget glial end effect. They could also be sensitive to those signals and mediate vascular effect. Okay, not only the signals, I mean the neurotransmitters which are released around the blood vessel may act on its wall, but also may act on these vascular glial end feet, which are very precisely located in the most deformable region of the perivascular tissue. Okay, if this interaction occurs, it would affect end feet first. Okay, and they can mediate, of course, glial cells can mediate and regulate and compensate or enhance the effects of contraction or dilatation. Okay, so we have here several possibilities of interacting with the vessel wall just because they move, and these movements, remember, are made to control local blood flow. So local blood flow can be regulated by synaptic activity, either directly or by the mediation of glial cells. Okay, please remember, which I forgot to say, that vascular changes happen in a scale of seconds or even minutes, while electrical activity of neurons and synaptic activity uses a very different scale. It happens in milliseconds. So synapses have time enough to interact with the vessel wall during these vessel wall movements. So this was published some years ago if you are interested in it, you can find it easily. And well, thank you very much. If you have any question.
Thank you, Angel, for a nice talk. Well, impressively, we have finished the talks at 1:20 as planned, so that's very impressive. And now we have time for questions and discussion. You can open your video if you want, and well, now we have some questions that have been sent earlier. In the chat, we have a question for Blanca, sent by Manuel Antonio Oria, and he says, let's suppose that you conduct parabiosis experiments in which blood from young mice is transfused in old mice and vice versa. Would you expect a change in mechanical and molecular properties of the transfused cells?
Well, I think we are not measuring, for example, the causation of the molecular biophysical changes that we are observing in the analysis that we are performing, in the sense that we are characterizing different parameters, but in the end we don't know, for example, if at the elderly age the, let's say, stiffening of the cells is produced by what. I think that is the well, that's the next, I think, perhaps biomolecular step. We could investigate more at the, for example, either genome data or some other more, I think, oriented, or I think this was more of an exploratory research project, and then probably some other hypothesis-driven experiments or projects could depend in the causation of the changes that we are seeing. However, regarding the question of whether the cells would change once injected in older individuals, perhaps no, in the sense that perhaps the phenotypical changes that we see by the mechanical stiffening, etc., it's given by the damage accumulated in the cells themselves. So perhaps there is a improvement in the health condition, since there will be more nice cells from young individuals. But I think with the information, at least of our project, we cannot reply with 100% reliability to this question. But why not? It's an interesting, I think, approach. Thank you.
Okay, thank you, Blanca. There is also a question for Angel Merchan, sent by Thomas Scheibel. Is the displacement of the synapses dependent on the vessel diameter? We cannot hear you, Angel. You need to.
Yes. Now you should hear me.
Yes.
Yes, of course. Different blood vessels would change their diameters very differently, and a single blood vessel would change its diameter very differently in response to different stimuli. Okay, because you can cause contraction, for example, with very different substances, and this will the power of these substances to elicit any contraction or dilatation will be different in the same blood vessel. This is very variable. Okay, we restricted our model to a 10 microns diameter vessel, because these are the what we call the pre-capillary vessels, which are the most, let's say, the most dynamic vessels regarding the control of blood flow in the brain. Okay, but you could extend the model to any other blood vessel, of course. and it's also very important. We made some simulations with different diameters of blood vessels. The conclusions do not change. I mean, the changes in the perivascular tissue are restricted very locally, okay? Very locally. Most of the cortical tissue is not affected by vessel movements. And this is important in both ways. There is no interference with most tissue, but this interference, if occurs, might take, may have something to do with vascular regulation precisely.
Okay, thank you.
Thank you.
Perhaps I'm not sure that we can see the hand icons. So if you may ask a question, perhaps you can directly open your microphone, because I'm not sure that otherwise we can detect the questions. In fact,
it's not working. This we have been checking that, and it's not working. So the only thing you can do is there is another icon in participants. There are like 3 dots. Maybe you can choose the applause icon, like I'm putting now. You know, and this seems like a hand. For some reason, it's not working this one. So maybe this is what, maybe, you know, if you want to do that, or just open your microphone and then you can moderate, of course. Just a suggestion. Sorry.
okay. I'm not sure if in the first talk, perhaps there was a question by Miguel Vicente Manzanares, but I am not sure, because I think we saw a hand. I don't know if he's here. Miguel Ángel, we cannot hear you.
Sorry, I have been talking to Miguel, and so then you could see the hand that at that point, but then after the break is not shown. So even Miguel cannot raise the hand now. So anyway.
Okay. In fact, I have a question for Fidel regarding your results. Could you talk about the physiological implications or your findings?
Oh, may I share the screen again? Oh, can you hear me?
Yes.
May I share the screen again? So
I think so.
Oh yes. Okay. Just hold 1 second. Can you see it?
Yes.
Okay. Here. So I think our discoveries can have many implications in mechanobiology in general, and I have here just put it 3 examples. Just think about integrin signaling. So there's been different publications saying that change in plasma membrane tension affects integrin activation. So if these Caveling-1 invaginations are able to sense very mild forces, the ability of these structures to flatten or not is going to directly affect plasma membrane tension and therefore integrin activity. And this can have massive implications in cellular migration and many different scenarios. Like, for instance, in tumor migration, I just found out, I came across this paper in which the title is PTRF, Caveling-1 expression decreases the migration of PC3 prostate cancer cells. So basically, if we just put this into the context of our discoveries, is that PTRF expression in these cells that normally lack PTRF, so they only have Caveling-1 expression, in normal situation, these prostate cancer cells would have Caveling-1 invaginations, but upon PTRF expression, most of these invaginations are going to be restricted to caveoli, and therefore the ability to sense different types of forces are going to be reduced to the thresholds to which caveoli are able to sense. And this is impinging in migration, so it's actually reducing migration. So I think this this could be somehow explained by our discoveries. And also some tumor stiffness sensing laws. There is tons of literature saying that most of cancer cells are basically losing the stiffness sensing. And here's just an example. Here you have fibroblast in this publication. You have normal fibroblast, and this is just durotaxis assay. Here's the stiffer part, and this is the softer part. You can see the normal cells, basically, as been shown in some of the presentation, that basically cells tend to migrate to the stiffer part. But if you can have different tumor cells that are able to migrate to the softer parts. And instead of losing stiffness sensing, what I think is that they are, because in this publication is actually one of the things they were analyzing, they were downregulating caveolin-1 expression in these cells. So upon caveolin-1 downregulation, one of the things that I think might be happening is that PTRF is released from the plasma membrane. So most of the invaginations that are going to appear in the plasma membrane are going to be caveolin-1 dependent. And these invaginations are sensitive to lower forces. So these cells, these tumor cells, rather than losing stiffness sensing, are actually gaining the ability of sense much more variety of forces. And that's the reason why they can migrate to the softer parts. So this is just 3 examples of the physiological consequences, but also one of the things that I've been, keeping thinking about is that caveolin-1 has been also discovered to be expressed in some neurons. And most neurons, or according to the literature, neurons basically lack caveoli. But if some neurons express caveolin-1, they could have, potentially, they could form these invaginations, enabling these neurons to sense very, very mild forces, which to me, it makes a lot of sense because the brain tissue, as Angel has said, is a very soft one. So cells within the brain tissue should be able to detect very small forces, no? Because this is the physiological context in which they are living. So I might imagine that it would be better for neurons to have, sensing mechanisms within the membranes that are allowing them to detect this type of forces that they are being subjected in the tissue. So these more or less would be some examples, physiological examples in which I think it could be have an impact.
Okay, very interesting. Thank you. Yeah.
If I may, that was very very nice. Presentation and answer by Fidel. But also, I think that one scenario where this those 2 systems of mechanosensing of different range of forces is could have a role is in the cardiovascular system, because when you have, for example, in different parts of the vascular tree, you have different types of blood flow regimes, and the forces are different. So maybe one mechanism is relevant for low range forces, these large invaginations that we have seen together with the group of Britta Qualman, or the classic caveoli that was described by Christophe Lamas and Vidisa Sinha, like 10 years ago, that is probably responding more to to high range of forces. Something that we are looking now into this, but this will be another potential physiologically relevant scenario. Thank you.
Okay, thank you. Okay, so if you have questions, you can use directly your microphone, please, because as we said earlier, it's difficult for us to see icons.
Gustavo, thank you very much for all the presentations. They were very, very, very good today. I had a couple of questions for Rebeca about her work on ER, on this ER shaper, the reticulon. The first one is how physiological is this kind of periodicity? It looks as if the reticulons were making like small domains within a tubule. Has this been described in vivo? And the other thing is that whether this will be because recently it was proposed that there's some kind of like peristaltic movement of the luminal content of the ER. Do you think this might have a kind of role for this, because it's as if it's connected to the microtubule system?
Okay. Well, regarding this peristaltic movement, I assume you mean in the lumen of the endoplasmic reticulum, of course. As our system in the optical tweezers is completely in vitro. It's a reconstruction of a membrane, but it's not from purified membranes of reticulum. It's a lipidic mixture with the with the purified reticulon. So it's a minimal system. So I'm not really sure that is completely related with the real physiological structure of the branches of the reticulum. So I don't think that these bulges and constricted areas are resembling the structure of the reticulum. And the other thing is that this periodicity, this kind of bulges and constrictions, as I said, in the pictures that I have shown, maybe it's somehow periodic, but it's not always the case. Sometimes you have a huge part that is constricted and then 3 or 4 bulges, you know. So it's just an stochastic feature, because reticulum has the tendency to oligomerize. So the parts that are bulges, we assume that there are oligomers of reticulum protein that are in the tube.
Yeah, thank you. Thank you very much.
Thank you for your question.
Okay. Thank you. Any other question?
Can I ask something?
Yes, Ignacio.
Yeah, I'm Ignacio from CENIC. It's a question for Fidel. Thank you very much, Fidel. Very nice presentation. So have you considered the possibility of this caveole stretching after tension as a way to expose some proteins that might be on the surface, I mean, on the plasma membrane, but kind of hidden from potential ligands. And then when the cell is stretched, then the protein may become available for ligand interaction.
Can you hear me?
Yes.
Okay. Yes, well, actually, there are some publications saying, like, for instance, with ion channels. This is something that has been, you know, shown that whenever caveoli flattened, you have opening of the ion channels. So, and in the case of, I remember I came across a publication saying that caveoli could be also considered kind of shelters, no, in which receptors are hidden and once, and they are protected from the external extracellular environment. So yeah, this could be one of the things that can be happening upon flattening, that some receptors are hidden and one caveole is flattened. They basically expose some domains that were not available, and then they can interact. And in the case of integrins, I think this could be one of the issues, one of the things that could be happening. Not that I don't know exactly if integrins are localized within caveoli. I remember I read something about that, but I don't think it's quite clear that real integrins are inside caveoli. But at least upon flattening of the caveoli, you are actually changing plasma membrane tension, and this clearly affects the activation of integrins.
Mm.
So,
Yeah, no, I was thinking more like a way for a very fast response. For example, in a situation where you have a ligand like abundantly present in extracellular space, but then just by like a quick stretch, the cell may be able to respond without the need of any protein production or even gene transcription or anything. So I was more thinking in
so how the conformation of receptor would be altered during. Oh, sorry. Seems to be some problems with your microphone, Nacho.
Can you hear me?
Yeah.
Yeah. Yes, I was thinking not the conformation, but the accessibility, just because maybe they are hidden like inside the caveoli, and then when when the membrane stretches, then they appear again in the surface and then become available. And I guess that would be especially relevant for big proteins, or maybe, I don't know, I'm thinking on LDLs, something related to lipoproteins, which I don't know if it could be relevant also.
Yeah, this could be, well, maybe the case, yes. Yes.
Okay, thank you very much.
You're welcome.
Okay, thank you again. Any other question? If there are no questions, in fact, it is 20 to 2. It was the planned time for finishing the seminar, so we are always on time. And well, if there are no questions, we can finish now. Perhaps Miguel Ángel, you can give some information about the continuation of this seminar series.
Okay, thank you very much. First of all, thank you very much, Gustavo, Pepe, and Maite, and Fran, and everybody involved from your institute and from the Politecnica and the Montegancedo campus for hosting this event. You know that, as you know, and for people that are not familiar with these seminars, it was thought to be actually physically celebrated in Montegancedo, actually in June. But we have to readapt, and as I was saying in the introduction, it will be very nice to take advantage of the positive side of this pandemic. And I think that by organizing this online, I think that we are actually taking advantage of something that is physically impossible when we have the physical meetings, which is to make them open to actually all the scientific community. And this is actually what we are thinking for the next one. So we are thinking again after Christmas to have another event. For this, we need people willing to share their work, like the speakers today. That I thank everybody for very nice presentations. Very high level. I was impressed by the scientific level of the talks. Congratulations to everybody. So then, as you know, we have this email list, Mechanobiology Athenic. It's a Athenic list, but it's an open list. So everybody from these other institutes that you already belong, that you can send an email if you have a particular interest on giving a talk in the next seminar. So you can send the email either to me or to Jorge or to Ana Gutierrez Llaneza, who is the secretary now that is handling this list. So please send us your interest in giving a talk. Looking at the pandemic situation, most likely the next event will be also online. If situation improves dramatically, positively, which I don't think is going to happen anytime soon, we will come back to the physical seminars. For that, of course, we need an institute that is interested in hosting this event again. So as we already have CMB and Universidad Carlos III, so Fernando in CMB and Arrate Muñoz in Carlos III, and now Gustavo and Pepe in the Polytechnica. So if any other group is interested in hosting, please drop us an email to either Jorge or me, and we will get this going. So we are thinking in naming these events. Before was the CENIC Mechanobiology seminar series, and now that we are opening to other institutes for the physical celebration, we are thinking in naming or getting this name of Madrid Mechanobiology seminars. But as I said, the interest of these are open to all the mechanobiology community, or all over the world actually. So this is the advantage of these online seminars. So let's take advantage of this while we have the pandemic, and then hopefully soon we can come back to the physical seminars. Thank you again, everybody, and also Jorge, that I think he was connected, at least for the presentation from his talk from María Rosaria, and then I think for some other talk. So I take this advantage to congratulate him, because he's become a father for the 3rd time, so everything is perfect, and everybody is healthy and happy. So congratulations, Jorge and family, and everybody again, thank you, and see you next time. We will advertise through the list and Twitter and the different channels, also in the CENIC web, so you can keep connected to these seminars. So thank you very much, and see you next time. Thank you, Gustavo and Pepe.
Thank you all.
Bye.
Thank you.
Bye. Thank you. Goodbye.
Thank
Bye.
you, everybody.
Bye.
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 Bye.